Welcome to Day 3 of the LGMD International Conference. Today's sessions will be filled with information we hope that you find helpful, no matter where you are in your LGMD journey. One key area of focus today will include the diagnostic process. Getting an accurate and confirmed diagnosis is very important for management of your health and for furthering research to find a cure and treatment. To begin our day, we have our platinum sponsor, SBio. They will be presenting an update on their upcoming gene therapy clinical trial for limb-girdle muscular dystrophy type 2I.
Hello, and thanks, Carol. My name is Anh Nguyen. I'm the therapeutic sector lead at SBio for the musculoskeletal programs. And thank you for the opportunity to present today at the Jain Foundation LGMD International Conference. And our discussion today is on the importance of collaboration with patients and clinicians in order to advance the future of gene therapy. Our session objectives today are an introduction to SBio's platform technologies, the science behind the Lion 101 gene therapy program, a clinician's perspective on the importance of academic consortia to collaborate with industry to advance new treatments presented by Dr. Nick Johnson at the GRASP LGMD Consortium, a family's perspective on being a parent to a family member with LGMD2I/R9, presented by Craig Norton, and a patient's perspective, being a person with LGMD2I/R9, presented by CJ Norton. At the end of the presentations, we'll have a panel discussion moderated by Dr. Sharon Heschley of the Muscular Dystrophy Association. And so before we start talking about SBio, we'd like to thank the Muscular Dystrophy Association for funding the initial research that helped the LGMD2I/R9 program become what it is today. So thank you. SBio, again, is a gene therapy company located in Durham, North Carolina. And what we do is we work with viruses called AAV, adeno-associated viruses. And we use these viruses to help develop medicines to treat potentially devastating diseases. The company was founded in the early 2000s. Again, it's located in North Carolina. We have a very strong discovery pipeline across many different therapeutic areas, ranging from rare disease, neuromuscular, CNS, cardiovascular, and metabolic diseases. We also have enhanced manufacturing facilities across the world and have a very strong intellectual property portfolio. As described about our pipeline, we have programs ranging from ultra-rare orphan diseases such as limb-girdle muscular dystrophy 2I/R9 up to very more common pathodiseases such as congestive heart failure. Our programs are in various stages of development, going from discovery and preclinical and into humans in phase 1-2 studies. For the limb-girdle muscular dystrophy program, we intend to initiate dosing for the gene therapy in the first half of 2022, next year. If you do have any questions about SBio and our clinical programs, please don't hesitate to contact us at Ask First, which is our collaborative program to help inform advocacy groups, communities, patients, their families on our research in gene therapies. And you can contact us at askfirst@askbio.com. In a moment, we'll play a video by our founder, Sheila McHale, on whom SBio is.
When somebody has a devastating disease, it affects not only the family structure, but it really affects the community at large. Disease doesn't just affect the patient, but it affects a much wider spectrum of people. SBio is an AAV gene therapy company. We provide treatments for devastating disease. Often these diseases have no viable treatment option. We have worked on Duchenne's muscular dystrophy, Pompe, hemophilia, heart failure, and Parkinson's disease. SBio was started when I gave a course at University of North Carolina. The course was teaching professors how to start spin-out companies, and in the audience was Dr. Richard Jude Samolsky. And he had a lot of intellectual property at the university. So he came up to me and he said, I need help, but I can't pay you. And there's a lot of skepticism about this technology. But if and when we make it work, we'll change the world. Gene therapy is a very simple concept. Now the question is, how do you get the gene into the body? We use an adeno-associated virus, or AAV, as a carrier to get into the body. So what we do is we take out the wild type DNA of the AAV, and so now it's just a protein shell, and in that we can put our therapeutic payload. And for most diseases, that means a one-time treatment option for patients. Duchenne's muscular dystrophy is a neuromuscular disease that affects young boys. These little boys lose the ability to walk, usually in their early teens. Often they die by the time they're in their 30s. Today, our drug is being advanced by Pfizer into pivotal clinical trials, and we've seen the results of little boys who were treated who can barely walk 4 steps and are now in swimming lessons and Little League baseball. It took us a long time before we got our first round of financing. Today, the company is over 350 professionals operating in 5 different countries. We have 3 different drugs in the clinic, and we expect that we'll have a very strong pipeline with many more therapeutics advancing into the clinic in the very near term. I carried a note for a long time from a little boy who I met who had Duchenne's muscular dystrophy. And so he wrote me a note saying, I doubt you're going to be able to have a therapeutic in time for me, but I'm going to look down from heaven and I'm going to be looking out for you, and you'll have a therapeutic someday for other boys. That note was really like the thing that kept me motivated. You know, we thought, and we were right, that we had the potential to treat these diseases. We just needed the opportunity to prove that we could do it. What I like most about being an entrepreneur is every day there's a world full of possibilities. You know, pick a disease, we'll work on it, and we'll make change. And that's incredible. That's really incredible.
Thank you again, Sheila. So when we talk about healthy muscle cells work by being connected to one another and to form muscle fibers and muscle bundles. And so this is important to have the individual muscle cells connected to each other so that when they get activated, they all contract in a synchronous motion and relax in a synchronous motion. And when the muscle cells themselves are not connected appropriately, they move not in a synchronous fashion, i.e. like in a not random, but sort of asynchronous way, and that increases the risk for the individual muscle cells to be damaged and have increased wear and tear. That degree of wear and tear is dependent upon what kind of defect there may be in the muscle cells themselves. Either it can be, for example, an enzyme problem, a structural protein problem, a channelopathy, getting nutrients in and out of the muscle cells, the contractile elements, as well as like sort of cellular repair mechanisms. And regardless of all of these different causes, ultimately it funnels down to, again, an increased wear and tear issue in the muscle cells that causes the muscular dystrophy. For the limb-girdle muscular dystrophies themselves, we know there are at least 34 different limb-girdle muscular dystrophies. All of them have different onsets, degrees of progression, severity, and that basically comes down to, again, what is the genetic defect? For us at SBio, we are developing a gene therapy program that focuses on an enzyme called a fukutin-related protein, FKRP. And this is an enzyme which is important to help basically the anchoring protein connect muscle cells to surrounding tissues and to other muscle cells. So as described before, at least 34 different types of limb-girdle muscular dystrophies. Again, multiple causes for why they occur, but what we do know, funnel looking in the big picture is that they're called limb-girdle because it has to do with large muscle bundles, which are predominantly seen where the appendages attach to the body, and hence there's weakness in the shoulder girdle, having problems like lifting things, moving your arms, as well as the hip girdle, which is important for core strength, as you know, getting in and out of chairs, bed, and just mobility in general. So as described before, the 2IR9 subtype of limb-girdle muscular dystrophy, it is a monogenic disease, a single gene disorder, for which there's a deficiency of the enzyme, the FKRP, again, which is important for the cementing of the muscle cells to each other and to form muscle bundles. And without this enzyme or sufficient levels of it, there can be progressive disability and death. And so what we do know also about FKRP, the FKRP mutation, is that there's many different reasons why you can have an FKRP mutation. There's at least 100 different reasons, different mutations. However, for the focus of our discussion today, we're focusing more on the common European mutation, which is called the L276I mutation, for which occurs from a gene mutation perspective. Either both the genes in human are affected, hence a homozygous mutation, or only one of the two is affected, a heterozygous mutation, which again is more rare. And the type of mutation is important because those with a heterozygous mutation tend to have an earlier onset, and in addition to that, more rapidly progressing and more severe form of the disease, in contrast to the later, more milder adult onset disease. But in any case, for everyone who does have LGMD2IR9, is that there's a significant burden to both the patients and the families and the communities themselves, for each person who has a 2IR9, there's a loss of productivity to the patients themselves, as well as their families for informal care, and then direct cost of care services. Again, not a significant, it is a significant burden to society. In about a third of the patients who have 2IR9, there is basically involvement of muscles of both the heart and the diaphragm that can lead to heart failure and respiratory failure. And so that this is in contrast to other milder forms of LGMDs. So, as described before, there are no therapeutic options, approved therapies for LGMD2IR9, and thus the predominant standard of care is just symptomatic management. So, knowing these details, we at SBio had went through due diligence to figure if there's an opportunity to provide a gene therapy. And so the notion around enzyme replacement therapies is not new. We've seen that with other diseases like metabolic disease, such as Fabry's disease, Pompe's disease, and that can a little bit of enzyme replacement or cause physiologic restoration of enzyme levels to help treat the disease, and thus our exploration into LGMD2IR9 as a clinical program. And so the Line 101 gene therapy program is again an investigational gene therapy for the treatment of LGMD2IR9 for both the homozygous and heterozygous mutations of the FKRP gene. And by being homozygous and heterozygous mutations, we intend to treat both the pediatric and adult patient community with this disease. And winding back to what a gene therapy is, there's different kinds of gene therapies. There's gene therapies, for example, where you take cells out of the human body, modify them, and return them to the body, and that's called an that's called an ex vivo therapy. We do an in vivo therapy where we provide the gene therapy directly to the human body. And this is done through, again, these AAV, adeno-associated viral vectors. And so what that basically means is that there is this virus called the AAV. It's known to not cause direct disease to human beings and has been used successfully in other gene therapy programs. For example, in Avexis's or Novartis gene therapy for spinal muscular atrophy. And so these viruses, these AAVs, what we do is we basically scoop out the internal contents and leaving a viral envelope, a viral shell. And we know these shells can be specific for tissues within the human body. And so, for example, in our design of our gene therapy, the virus envelope, we believe, has a preference for the muscle tissues, right? And we've seen this successfully in peer-reviewed literature for other clinical programs. In addition to that, with this virus shell, the scooped out contents, we put in basically a healthy copy of the FKRP gene with something called an activation sequence or promoter to help support this healthy gene to be produced in a sufficient number of copies of the muscle cells. And so again, virus shell promoter to help activate making copies of the gene and the healthy gene itself. So the viral shell is specific, we believe is preferential for muscle tissues. The promoter, the activation sequence for the healthy copies of the gene has been designed to be muscle specific. So in other words, when this therapy is given to the body systemically, we believe, and again, it'll be evaluated in clinical studies, that the healthy copy of the gene will be most expressed in the muscle tissues. Again, the heart, the diaphragm, and the skeletal muscle. Because of the design, unique design of our gene therapy, we believe that we will have a favorable benefit risk evaluation or assessment where potentially we can get the effects we want, the benefit we want from the therapy at lower doses of the gene therapy, thus reducing the risk of any sort of side effects. We have done so far preclinical studies in animals with LGMD2I/R9 to evaluate both the toxicity as well as the appropriate range of doses we want to give the gene therapy. And thus far, they've been pretty promising. So from a safety profile, we've seen very little issues around the side effects for the disease. However, we do have to evaluate that in human beings. At the same time, we for the efficacy in terms of, for example, seeing muscle force, endurance, that compared to an animal model with the disease without therapy and a wild type healthy normal animal, like with the gene therapy, we essentially have near normalization of muscle force and endurance. Again, physical function returned back to the animal model, approaching that of wild type mouse. Again, very, very promising. And so thus, we believe that we've identified a potential gene therapy that could provide that favorable benefit risk assessment and in a dose range which we believe would be safe for human beings. And thus, we are in the process of doing human studies. We were authorized by the FDA this past year to initiate first-in-human studies, which we believe we'd like to dose in the first half of 2022. And so this is sort of a higher level schematic of our clinical program design, where we basically are here where we will evaluate the recommended range of doses for the gene therapy to see preliminary safety and efficacy of the gene therapy, assess that the therapy is working appropriately, you know, in the muscles, and based upon these results, finding the appropriate dose, we find the best trade-off for safety and efficacy, we move into a larger program to confirm the safety and efficacy of the gene therapy, again, for purposes of the marketing submission and approval. What we can share is that the design right now is a double-blind randomized placebo-controlled study, which means that what patients who are eligible and enrolled in the study will receive the gene therapy or a placebo. And this is in order to find the most robust design to see, again, to evaluate safety and efficacy, and then to move forward through the program. At the end of the respective sections of the study, patients who are randomized placebo, given that they remain eligible, will be offered therapy with the Line 101 investigational gene therapy. So in other words, for the entire clinical program, again, given that patients remain eligible, that they will be offered an opportunity to receive the Line 101 gene therapy program, or gene therapy. So if there are any questions regarding the clinical program, please don't hesitate to contact us again at AskFirst, our patient engagement platform, at askfirst@askbio.com. And so thank you again for the opportunity to present at the Speak Foundation LGMD International Conference, and we look forward to further presentations today. Thank you.
Hi, everyone, and thanks for having me. My name is Nick Johnson. I'm Vice Chair of Research in the Department of Neurology at Virginia Commonwealth University, but really coming here as a PI of the GRASP LGMD Consortium with an update from the consortium. Here are my disclosures. So I think it's always helpful to cover a little bit of the background here. Specifically, I think everybody here knows the limb-girdle muscular dystrophies collectively represent a group of disorders that cause progressive weakness that most affects the shoulder and hip girdle, but really can affect any muscle in the body. That progression is slow, but it is progressive, and that progression is meaningful. And eventually continued muscle damage may lead to replacements with fat and scar tissue. And that's, you know, at the end of the day, some of these exciting therapies that are coming down the road, as you heard about, are designed to prevent that from happening with any luck at all. So I'm here representing the GRASP LGMD Consortium. And really the mission and vision of our consortium is to accelerate the development of therapeutic trials and to make sure that those clinical trials are successful. And what is a successful clinical trial? That means that we're measuring something that's important to people with limb-girdle muscular dystrophy, that we measured as precisely as possible and able to really say whether or not that therapy is working. So what are some of the challenges that we have? Number one, there is limited knowledge of the natural history of limb-girdle muscular dystrophy. That may be surprising for those of you who've been living with LGMD for your whole life, but there's been very little in the literature written about many of the different subtypes of limb-girdle muscular dystrophy, or measuring how that progression might occur over the course of 1 to 2 to 3 years. And along with that limited knowledge of natural history, there's been limited development of clinical trial tools. These are the kinds of things that we need to measure that progression, measure the change in walking or your ability to go upstairs. And then lastly, intrinsic to the disease itself, the slow progression of limb-girdle muscular dystrophy means that clinical trials may need very sensitive measures or many individuals to enroll to show change in the treatment. If you think that on a given outcome measure that the average rate of change in limb-girdle muscular dystrophy is about 5% per year, then in order to show a difference with therapy, that may need a large number of people to be able to participate in the clinical trial to show a benefit, or we may need to come up with better ways of measuring progression, as we'll talk about. So where are we now? I mean, I think this picture is classic, and I'm excited that the rabbit, which is therapy development, is ahead of how prepared we are, but it does mean that we need to catch up. I mean, this has got to be one of the more exciting times ever in the field because of the amount of amazing development of these gene modifying therapies and other approaches. But we're not ready for it. Our trial preparedness is we're not where we need to be. We don't have as good of an understanding of the natural history as we need to, and biomarker development has lagged as well. So these natural history studies, such as the ones being conducted by GRASP LGMD, provide the tools for clinical trials. They look at outcome measures that tell us whether or not something has changed between 2 points in time. They also look at biomarkers, which are something that you can be measured in blood or muscle, a sign of how the disease is occurring. And they might be looking at something like an evaluator assesses like strength or the ability to do tasks or even questionnaires. And, what's the sweet spot? Our outcome measures should be pertinent for you. They should be reliable. They should be sensitive to change, inexpensive, and able to be done across the world. And we have not settled on the right mix of outcome measures yet. One key cornerstone of how we're thinking about therapeutic trials is mounting these clinical outcome assessments to find that sweet spot. We're looking for that orange line. So we're looking for something that changes a bit more than what we would predict across the whole population. The blue line shows something that doesn't change much at all. And the gray line shows something that might change too quickly, that the therapy might not have an opportunity to affect. And so we're very focused on understanding prior performance in an outcome measure and seeing whether or not that may predict future performance. And we continue to work on developing outcome measures that capture symptom outback. Biomarkers. So beyond the clinical outcome measures, a word or two about biomarkers. So for gene therapy replacement, the obvious biomarker is expression of the missing protein, similar to what they use in DMD. For some limb-girdle muscular dystrophies, it may be other things that we measure in blood, such as evidence of muscle destruction or inflammatory markers, or even muscle MRI. So this is an MRI, and you can see up in that panel the little percentages showing how much fat has come in and replaced that muscle. And that's the kind of thing that may be a sensitive biomarker that can be used in a disease like limb-girdle muscular dystrophy. But there's a lot of work to be done. We have the advantage of borrowing heavily from fields like Duchenne muscular dystrophy, which is a little bit ahead of us, and we can learn from their both failures and successes. But we still have a lot of work that needs to be done to ensure the biomarker is reliable, that they're responsive. And then, the key question is how much protein needs to be restored to be functionally relevant. That's really an important question, and not what we won't know until we engage in clinical trials. So here's the GRASP LGMD studies. It's a platform natural history study, same outcome measures, same sites, same training. And this is our ability to study new subtypes. And we're focused on developing outcome measures for upcoming clinical trials. Most of these studies are at least 12 months in duration, if not a little bit longer. And they all include surveys. They all include measures of muscle strength and functional testing, a blood draw, and occasionally a muscle biopsy or MRI. You can see there the current GRASP LGMD studies. It's a little bit of the LGMD alphabet soup, ranging from 2A, 2B, 2C, 2D, 2E, 2F, 2I, 2L, and D1. And the sponsors are there on the far right side. We couldn't do this, particularly calling out the NIH and the MDA for really being some of the Coalition Cure Campaign 3 is for being some of the original sponsors. These are your current consortium members. Look through that list. You might be able to find a site that's close to you. If you can't, in some cases, we're able to provide funding and reimbursement to get to one of these sites. And of course, these are we're proud to have this group of investigators because these are top-notch minds really focused on developing therapies for limb-girdle muscular dystrophy. So please consider enrolling in a GRASP LGMD study. That's my email right there. Or you can contact Jessica St. Romain. Her email's right there. And we're happy to talk to you about it and see if we can't get you to a study or site that's open for you. These natural history studies prepare the community for the disease-modifying trials. Our ability to successfully execute the exciting therapies such as what Dr. Nguyen was mentioning really hinge on our ability to inform such trial designs and make sure that they're going to be successful for you and for the rest of the community. So these upcoming trials really offer potential benefit. To close out, promising therapeutic development using gene replacement technology across the limb-girdle muscular dystrophies really has hastened and focused our efforts, sped our efforts in some ways, and that these natural history studies are essential for appropriate trial design. And our consortium is performing these studies in multiple subtypes. And we hope that if you're listening and you have one of those subtypes, that you would consider enrolling in one of these studies. Thank you.
Hi there. My name is Craig Norton. This is my wife, Jody Norton. We live in Northern Virginia. We've lived here in actually Fairfax, Virginia for about 3 years. Originally from Arizona. Both of us were raised in Arizona and had both of our children in Arizona, same hospital as a matter of fact. And they lived in Arizona up till age 12 and 10 respectively. So we've transitioned away, and the reason I'll go into that a little bit more is as we talk about the disease, and we'll probably start chronologically. When we found out our first child, Brody, had it, the first inclination that we had that anything was wrong. First of all, no family history that we know of. Neither one of us, both of us relatively active. I'm in the Army and have, in a relatively physically demanding career field, and Jody at the time was teaching a fitness class. So from a muscular dystrophy standpoint, that was really something that we hadn't envisioned or seen or had any relatives that we know of that had the disease. So we initially, our oldest was 15 months. His neck sort of went limp, and took him back and forth to the pediatrician several times. They couldn't really come up with what was going on with it. And then we ultimately ended up in Phoenix Children's Hospital. They ran a battery of tests in which they determined that his CPK levels were through the roof, like 20 to 50 times what they should have been. And so then at that point, they started suspecting muscular dystrophy. He eventually, in that initial onset, recovered, and we were really hopeful that it was just an off virus that caused it. And so we moved on. I actually deployed to Afghanistan. At the time, my wife was pregnant with our second, and pretty normal. His progress didn't really, maybe a little slow to do certain things, but generally speaking, nothing outlandish. And so fast forward, maybe a year from that, 9 months, and the results came back that he indeed did have a form of muscular dystrophy that we'd never heard of, limb-girdle muscular dystrophy type 2I. And
that was after the muscle biopsy.
Yeah, they did do a muscle biopsy, and that came back as such. And so Brody was very small when he was a baby, and progressed smaller. And then his brother Conrad, CJ, came out, and he was just a big ball. And so not having much understanding of the disease or how it presents, we made the assumption that, he's big, and so probably the odds of him having it as well were probably pretty slim. So as that moved on, CJ was slower to do most things, crawl.
Yeah, he missed most milestones. He was a delayed walker, never crawled.
Yeah, and it was obviously on the brain, so we tested them, and it came back that both of them did have it. So moving on, as it is a progressive disease, they were, couldn't run as fast, couldn't really run, but could move out pretty good as they were younger, and that's when most of their childhood friends were made. They were able to do, flips on the trampoline and almost, most things. They were a little weaker, but generally speaking, they could keep up with their friends pretty well. So moving forward, we could probably go to that next slide if you wouldn't mind, Matt. So this was a couple pictures of recent. The one on the far left was swimming with the Dolphins in Atlanta last winter or 2 winters ago. Going forward, the transition to this area was a little bit more difficult because of some of the things that we probably progress one more slide. So, I'll just speak to the idea of being able to they look typical, but as we've gotten here, they've gotten, slower, and their limitations are becoming a lot more presented, and they're starting to not be able to do the same things with their peers that they were able to do, and it's becoming the chasm between what they can do and what their friends can do is becoming a lot greater. So moving to a new area where you're not able to do a lot of the things that kids bond with, it's been a little bit more challenging to find friends, and so their bonds and friendships, they haven't been able to create the same ones that they had at home, just because they have a lot of those shared experiences growing up. So that's just been a challenge with moving, that, potentially, if there were the sports activities that they could do, swimming, all those things that they're getting weaker and weaker, and like I said, the gap between them and their peers is greater, and so it's starting to create a lot more anxiety. I would say when they fall, it's hard to see as a parent, but when the idea of them being left out, and the emotional piece is really what eats me up inside, is when they feel as though they're I've never heard anybody make fun of them for anything, but I know they're self-conscious if they're walking a little slower or that sort of thing. So that very first bullet, it means that we hope and pray every day for a cure. That's from the very start. We initially started going to a neurologist in Arizona, Phoenix Children's Hospital, and very upbeat neurologist, and he wasn't a specialist in muscular dystrophy by any means, but whenever we showed up, he was always telling us how optimistic he was about the future, and just all of the interchangeable or inner disciplines that are leading to advancements in a lot of different disorders, and just the time period we're in today of advancements, and how that he was extremely optimistic that there was going to be something on the horizon. So when we got here, we were able to get into the Johns Hopkins program, which included, at the time, it was Kennedy Krieger, which was Dr.
Catherine
Wagner, who had very extensive background. And so very hopeful about that. She probably 6 to 9, I don't know, maybe a year into going to hers, when she mentioned the potential of the SBio gene therapy study. And really, since we heard about that has been the biggest groundbreaking in our mind, potential for changing their lives forever. So that's been foremost in our mind for something that we think truly has the potential to have a dramatic change. So I don't know if, Joe, do you want to talk to some of the things that just communicating with school and staff and.
Oh, well, yeah. I mean, as Craig mentioned, they do look like, your typical kiddo, with the exception that they are unable to participate in most sports. And we have to have, 504 plans and IEPs, set up so that all of their next year's teachers are aware of the fact that they may have to take the elevator. They may be a few minutes late because it may take them a little bit longer. What are we going to do about PE? Because they're not going to be able to participate. So in fact, we just left the high school, walking around with Brody and trying to, anticipate, plans of action and how he's going to get from one class to the other, and making sure that the counselors and everybody else is on board. And most people are, very accommodating and very kind, but it's just one more obstacle that we have to overcome. So I know that there's a lot of anxiety for both of them with attending school, especially after the last year that we've had with the virtual learning. So it's going to be an interesting year.
And with the fact that it is progressive, and I'd mentioned, when they were younger, they could do flips and, they were all over the place. We also, as we moved out here on Facebook, we had linked up with the Cure LGD2I. I saw it was, Kelly Brazo. And so she mentioned the Iowa annual natural history study that they do, and it's a conference. And so, for the first time, we'd really, with just the two of us, we didn't really know anybody else that had the disease or was afflicted, or any parent. So I know Jody for sure felt great to just be able to talk through some of these issues with other parents and other people that had gone through that. What was a bit of a unintended consequence was the first time we went to it, CJ, and going back to the theme that the kids appear as typical as possible. They, you can see in their gait they're a little bit different, but when we went up into the hotel room and CJ looked down and saw a lot of the older kids, in wheelchairs and scooters, I think it was a real, it just hit home that the potential that would potentially be his future. Just really, he saw that, and it was not the reaction we were going for there. So keeping that in mind, he's very, when we talk about accommodations for Jim, because they do appear as typical, to be sort of put in the spotlight that they're unable to do things, and hey, CJ, you're going to sit on the side while everybody else does these things, and it's explaining to the teachers that we do physical therapy. They're getting enough physical activity. PE might be something that's great for the typical kid, but in this case, going to the library might be better from a mental standpoint, that they're not going through that stress of being singled out, which nobody likes anyway, especially 13 and 16, and then to put on top of it, the concern they have of, hey, what's wrong with you? Why can't you do this? So just another obstacle that they deal with. And again, it sounds like a lot of our parochial issues are very centric to us, which I apologize for. But this is just specifically their situation and the progression of their disease. And I know LGMD has a wide variant, and I know it affects people at a much different rate and a pretty vast age range from when it hits them. But this is obviously for theirs, even between the two boys, between Brody and CJ. CJ's always been compensatorily weaker than Brody is, even at each age. So, and he seems to be progressing a little bit more rapidly. So, I think did you have anything else you wanted to talk to on that one, Jim?
I don't think so.
Okay, Matt, you can go to the next. So, as mentioned, we have taken part in the natural history study, but that's really the only clinical trial study that we've been involved in. So don't really come at this with a lot of experience, but sort of go down what to consider. we are very obviously foremost in our mind is safety. Don't want to do anything that's going to obviously harm them or
have any negative side effects,
impacts at all. So that's clearly something that we're mindful of. Very interested in the timeline that this particular study is going to be able to go from adults to children. So any impact or influence that you might have on expediting that is really what we're hoping for, and that leads into that fourth bullet of time is not on our side. As this is a progressive disease, every day that goes by without significant intervention is going to reduce the promise of sustained mobility. So, to us that means, and we, obviously we don't know what the full potential of the treatment might be. Is it something that's going to allow them to maintain where they're at? Is it going to be something that's going to reverse the progress of the disease? Or is it something that's just going to slow the progress? And we don't know, but we do know that the sooner that they're introduced to it, it's going to be able to have the greatest impact. Include us in the schedules, those are kind of self-evident. The other piece that I'd mention is I know as they go through studies, there's certain endpoints that they're trying to realize and assessments, and it certainly makes sense that you would look at anytime you're trying to measure anything, a before and after in order to demonstrate success. So with that, because this disease is so variable, there are many factors that I think still could be considered without, maybe being able to get up for a flight of stairs or off the ground without any assistance. So the wider range of possible measurables would be where we'd really appreciate. It all comes down to the fact that things that the kids could do 2 years ago at a certain rate, they're probably not able to do at that same level today. And so if there were other things that could be looked at and they wouldn't be excluded just because they're unable to do some of these things, that obviously being excluded from it would compound the problem even more, because they're on a trajectory right now that's going the wrong way. So and then that final one is hope. As mentioned, since we found out about this potential, it's been the most hope, and we've from our internal family to our extended family, that is, this potential is the source of multiple conversations, dinner table, bars, everything. So the promise that this holds has been one of the greatest sources of hope that we've had over the last 2 years since we heard about this, the potential for the gene therapy.
Hi, my name is CJ Norton, and I'm 13 years old, and I have LGMD type 2. And ever since, next slide. Okay. It means that every day, or ever since I heard about a cure, a potential cure, I've been praying and hoping that this cure can come out and work. It means it's much harder to partake in activities that all my friends can do, and I have to think ahead in every way possible. And one example today that happened just a couple hours ago is that me and my friends wanted to go to somebody's house, and we had to jump over a fence, but I wasn't able to, so we had to go the longer way. And it means I have to think a lot farther ahead of everybody else, and it means that I have to
well, it means sometimes that you're distracted on, if you're sitting in class and the teacher's talking and it's coming close to the end of class, things like, am I going to be able to get out of the seat? Is everybody going to walk by me? Things like that are more foremost in your mind, as opposed to, what you should be learning. And so, one of the things that you've worked on is trying to communicate that as early as possible, right? When you find out about things like, when you see that, let your teachers know, so that you're really advocating for yourself, right?
Mm-hmm. Yeah. So it means I have to tell people things that I don't want to tell them. Like if I'm 10 minutes, or if I meet them and they like me or something, and I have to tell them that, like, I have this LGMD, that, like, they'll stop being friends with me or something like that, because I don't know, they just think it's weird, I guess. But I also have to tell the teachers about everything, and if I don't have to, or I don't want to do something, I have to tell them about it, and it means I have
You have a lot of people staring at you and you feel like you're the spectacle. Yeah. Yeah, and that's where he's, because you're so likable, I think you usually overcome that. But with new school starting and everything else, he's knows a lot of the people, but last year was sort of a throwaway year as far as he's going to middle school, so it's 7th and 8th grade here. So getting to know everybody. Last year from his elementary school, it sort of increased. So while he does know quite a few people, there's people that he doesn't know that you probably don't feel as comfortable around yet, right? So anything else you want to talk about, maybe with PE and sports?
Yeah, in PE, I usually have to just sit out, and people ask, like, oh, why are you sitting out? Like, there's nothing that I could see that's wrong with you. And I don't like explaining it to them.
Yeah. Yeah. We, 2-3 years ago, we did, we had quite a run of flag football when they were younger. We ever won the championship?
No.
Yeah. Well, CJ had great hands. He didn't exactly tool down the field as the fastest guy, but he was like he was like a magnet when you threw the ball to him. So, but loves watching sports, and he always says to me, if it wasn't for this, I'd be the best athlete in school. So. All right. Anything else on any of that stuff, CJ?
I don't think so.
Okay, next slide then, Matt, if you could. So a lot of this is reiterating what we'd mentioned in previous slides, but I think the biggest takeaway, again, this is us, is I know a lot of the different treatments require travel. They require funding to get there, and I know in many instances the ability to compensate for that is highly beneficial. Specifically for us, if there was a treatment in Egypt, we'd find a way to get there. But so we because of what we feel the stakes are with this potential treatment, we would be at anybody's disposal from a scheduling standpoint. And we also just want to take this opportunity to thank everybody who's taken any part in potentially coming up for a long-term cure, or at least a therapeutic that's going to help enhance quality of life and sustain mobility for any that longer than the disease would naturally progress. So I think that's all we have. Do you have anything else, CJ? You want to thank everybody for all the help that they've provided and the work they're doing?
Thank you.
Okay, well, I think that's all we have. Thanks.
Hello, everyone. So I'm Dr. Sharon Hesterly. I'm the Chief Research Officer at the Muscular Dystrophy Association, and I know you just heard some great presentations from our speakers. Fabulous job. And now we're going to have an opportunity to do a little Q&A with each of them and sort of delve into some of these topics a little bit more. So maybe before I start, I think we'll start by introducing the panel. You've probably given a brief introduction, but let's start with Anh Nguyen. If you want to say a little bit more just about your background. How did you get involved in research and what is your motivation?
Well, thanks, Dr. Hesterly. And so well, first and foremost, we'd like to thank the Muscular Dystrophy Association for its support of this gene therapy program from its initiation and funding in the initial research and the preclinical research to get us to where we are today. So by background, again, my name is Anh Nguyen. I'm the clinical development lead for the LGMD programs at SBio. So by background, I was by training a physician scientist at the NIH, and I've also worked at the Food and Drug Administration, also at Medicare. So fundamentally, as a patient-focused drug gene therapy company, SBio has a lot of opportunities to be engaged in the community and in gene therapies for rare disease. And we do feel that this opportunity, again, to be part of the SBio Foundation conference is instrumental to us to creating better therapies. So thank you.
Great, thanks. And Dr. Johnson, do you want to comment a little bit about your career? You've been involved in neuromuscular disease for a long time. So.
I have, thank you. Yeah, so I did my training in neurology and then neuromuscular experimental therapeutics at the University of Rochester after getting my medical degree at the University of Arizona. And my focus since that time has really been on genetic nerve and muscle disorders. And in my role at Vice Chair of Research and Associate Professor at Virginia Commonwealth University, I've been fortunate to be the PI of our GRASP LGMD consortium, sponsored originally by the Muscular Dystrophy Association. So thanks for that support. And I've been excited to see that original small consortium grow from the 6 members into 14 centers at the United States and Europe, all dedicated and focused to research and development, accelerating therapies in the limb-girdle muscular dystrophies.
Great, thank you. And then Craig and CJ, is there anything you want to add, maybe that you didn't get a chance to add in your earlier presentation?
Well, the only thing I think we're really missing is just thanking the Muscular Dystrophy Association for the word that they've spread and continue to do. We're just happy to be a part of it. Good opportunity, I think, for CJ to be able to open up and tell his story and sort of have the opportunity to open up and I think it makes the hope a little bit more real to be involved in an opportunity like this.
Well, that's great. It's really gratifying for me to see this research come this far too, because I worked on this project previously when I was at SBio, and actually, I'm not sure everyone knows that MDA was an angel investor in SBio back in 2004. So it's really nice to see where the research has come sort of full circle here. So, okay, I want to start a little bit maybe with Anh. And, there are a lot of different ways to go about gene therapy, and it's still relatively new field. Can you tell me what makes you think, what's special about the approach that SBio is taking? Why do you think it's likely to be successful in this case?
Understood. And thanks, Sharon. And so on a high level, there's different types of gene therapy, right? And there's gene therapy that you can deliver directly to human body versus gene therapy, which you have to take, for example, cells out, modify them, and return them to a body. And so SBio specializes in vivo gene therapy, which means we deliver the gene therapy directly to the human body, either through different routes of administration. Specifically for the limb-girdle muscular dystrophy program, what we provide, we intend to provide is a single shot infusion of medication that gives systemic delivery to all the target tissues, i.e. the muscle cells within the body. Our gene therapy, again, is focused on expressing the normal enzyme and hopefully physiologic restoration of enzyme levels to the target tissues, i.e. muscle tissue, striated muscle tissue. So this would be skeletal muscle of the limb girdle, your shoulders and your hips, as well as potentially physiologic restoration of enzyme levels to muscles of the heart and the diaphragm. We believe that with the technologies that we provide would be complementary to what's out there right now, as well as an opportunity to provide, again, physiologic restoration with durability of effect, which means it could last, ideally for the lifetime of the patient who receives the therapy. In addition to the technology, with the promoter technology, we believe we can provide probably potentially lower doses of medication of the gene therapy, which in our field, as we know, that less is more, i.e. if you give too much of the therapy, you can have some side effects. And so we believe that we'll have to evaluate it in a clinical study, but that the right trade-off between the right amount of help and the lowering amount of side effects. So.
Okay, and then a quick follow-on question then. Why 2I/R9? Can you give us an insight into how you came to this indication?
Understood. And thanks, Sharon. It's really kind of a twofold kind of response. First is Asclepius Bio, SBio moved into musculoskeletal diseases. There were really two programs originally. One of the programs was in Duchenne's muscular dystrophy, which was part of a company named Bamboo Therapeutics, part of the SBio sort of umbrella. And that program in Duchenne's muscular dystrophy eventually moved to Pfizer. And so therefore, as we mentioned in our presentation, the viral envelope that's used within our program in LGMD2I/R9 is basically the same envelope used in the Duchenne's program. And that's essentially, again, SBio technology. The parallel program, and specifically in 2I/R9, the rationale is that we first wanted to move into limb-girdle muscular dystrophies, which were more common. And as you sort of look at the scope of the limb-girdle muscular dystrophies, there are I believe about 7 of them which cover the overall majority of the limb-girdle muscular dystrophies, more than 70%, of which 2I represents probably, I think it's maybe number 3 or number 4 on the list. And so we felt that was it was a disease, a rare disease for which there were a sufficient number of people who may be interested in receiving the therapy and us developing the therapy. In addition to being prevalent enough, is that our program, as discussed in our presentation, restores physiologic levels of an enzyme, right? And so enzyme programs historically have been very successful. So for example, you may have heard of enzyme replacement therapy for Pompe's disease, for Fabry's disease, and they can be done successfully because you don't necessarily need a ton of enzyme to really help the patient, right? And so we believe that using this with our therapy, we can restore adequate levels of enzyme again to help the patients. And as discussed in our presentation, at least from a preclinical model, the mouse model, we're seeing essentially, again, we have to evaluate it in human studies, but essentially normalization of muscle force and muscle function, endurance. And so I think was that helpful in terms of response? Or?
Yeah, no, I thought that was great. That made a lot of sense. So I guess my next question is for CJ, if he's willing to tell me. So, considering being first in something, being in a clinical trial, maybe for a drug that hasn't been tested in humans before, it's a little bit like being an astronaut. I'd love to hear how you feel about it. Is this something you're excited about doing or just really motivated to do? You want to tell me in your own words?
Yeah, I really, really want to do it. I'm super excited about it because of the benefits of it, obviously. And at first I was kind of scared because it's like a shot they put into you that nobody's ever really tried before. I'm like, I don't want to do that. I don't know what's going to happen to me. But now that I know a lot more about it, and I know a lot more about it, and I'm not as scared, I'm super excited about it.
Yeah, I would love to introduce you to a friend of mine sometime, Donovan Decker. He was the first person in a gene therapy study for Limb-Girdle 2A back in 1999. So it wasn't just the first gene therapy in a limb-girdle, it was the first gene therapy in any form of muscular dystrophy. And I still talk to him weekly. So I'd be happy to introduce you. I think he would probably have some interesting insights for that. That was almost 20 years ago now.
What were not to take too much time, what was it effective?
Well, he was, this was a true experiment. So it was just an injection in his foot muscle. So this was a completely altruistic act on his part. He knew he wasn't going to get any benefit from it. But it really, gave us a lot of confidence that we could do this in humans, and it moved on. It really was huge for the field. So in this case, now when we treat people, the goal is to, try to have a benefit if possible. So that's why we do the systemic delivery. We're not just putting it in a single foot muscle, but no, that's great. So now I'm going to ask Dr. Johnson, if you could give me some thoughts about this consortium approach. What do you think the advantages are? I know probably what the disadvantages are. It's a lot of work. It's a lot of cat herding probably to get all of these sites and all of these different people moving in the same direction. And you think, tell me why it's worth it.
Well, I think the consortium approach has several advantages. Number one, the clinicians involved, first of all, are not like herding cats. These are all really outstanding experts who have a singular focus on advancing therapies for patients with limb-girdle muscular dystrophy. So it's actually been honestly a lot of fun to work with my colleagues and work in pulling all in the same direction. So it's part of what makes my job so easy. The true advantage to the patients and to the research community, though, is that we've been training for this moment. Our sites have been trained on the protocols that would be involved. The clinical evaluators that are in the consortium have a massive amount of experience. And we think all of those attributes really not only will accelerate our ability to get the trial up and off the ground, but also to ensure the successful execution. As both you and I have known and seen development programs move forward in the muscular dystrophy space, it's amazing how much some of those kind of late stage hiccups can happen with equipment failures or lack of training and expertise. And so we really think that this approach prevents some of those late clinical trial pitfalls by having everybody, like I said, trained experts in their area and geographically dispersed to be able to provide trial sites to patients close to where they live.
Right. If I may add, and thanks, Dr. Johnson, is that fundamentally we're so grateful for clinician scientists like Dr. Johnson, because there's fundamentally creating new science is just from bench to bedside is just not easy. And as in patient-focused development for therapies, the industry sponsors intimately tied in with the perspectives of the investigators, of the clinician scientists. And so we're learning every day on what is standard of care, what is state of art, and how the state of art changes over time. In Dr. Johnson's discussion around clinical endpoints and biomarker development, as Dr. Johnson is the lead investigator for the GRASP LGMD Consortium, without the work of the consortium, the work of us at SBio could not exist. And so, and every day we're very grateful to learn from you and your organization.
Thanks. We appreciate it.
It's true.
Happy to help.
Yeah, and one follow-on question for Dr. Johnson. So what do you think the biggest challenge is in developing therapies for a disease like limb-girdle, which is more slowly progressive compared to some other forms of muscular dystrophy?
I mean, I think at the end of the day, that's really where the challenge is. And, when you think about developing a drug in a rare disease, you need to be able to detect change over time. And for a condition like limb-girdle muscular dystrophy, which is progressive, but slowly progressive, there can be quite a challenge in figuring out how to see that change in as few patients as you need to to enroll in the clinical trial, because one of the concerns, obviously, it is a rare condition. We don't want to overburden patients or subject people to placebo, for example, if we don't need to. And so we've put a lot of time, effort, and focus into understanding the phase of the disease across the limb girdles, where you might be able to see change in as short a time as possible to create an effective study. And that's really been largely around our how we watch people walk, for example, or climb upstairs and other things. And we think that that's kind of the sweet spot. But not to forget all of the important work in the biomarkers, because one of the nice things about limb-girdle muscular dystrophy and LGMD-R9, for example, is that you should be able to see restoration of function of that enzyme, and that provides a lot of hope and optimism early on in a clinical trial that we're on the right track, we have the right dose and everything else. So I think to me the biggest challenge is the slow progression of the disease, which is great as a clinician and when I treat patients in clinic, but does make the trial design a little bit more challenging than some other forms of muscular dystrophy.
So you're saying you could use the evidence that the enzyme activity has been restored as sort of a leading indicator. So you'd have some idea earlier that you're at least, this gene is doing what it's supposed to do, the one that you've introduced.
Yeah, I think that as trials develop across in LGMD-R9 as well as across the limb girdles, you're going to see a lot of designs that focus on early measurements of the protein function or presence, and followed on by later evidence of clinical benefit. And I think that combination does create a compelling story that the drug is doing what it's supposed to be doing.
Yeah, I think this is really great, exciting, progress, I feel like, in this area. And while all this exciting progress is happening, I think, CJ, you're still living with the disease. Do you want to talk to us a little bit, and maybe Craig as well? How do you explain this disease when you talk to family and friends? Have you come up with a way where you feel comfortable making people understand what it's really like?
Usually when I tell my friends is I have a muscle disorder that makes me not do as much as I can physically. Mm-hmm. And that's basically what I say to them. And
what do you think they understand? What do people out there understand about muscular dystrophy? Not very much.
Yeah, I don't think they understand.
Mm-hmm.
They understand that they need to help me sometimes, but they don't understand what's really happening with the disease.
Yeah, I think that's the challenge with having a relatively rare disease, is you're constantly having to explain it. I know people frequently respond when I say I work in muscular dystrophy, they say, oh, my aunt has MS. So pretty used to that one. Don't hear the words at all. Yeah, so I think that's a challenge. So I think we just have a few minutes left. I think before we go, I will just turn it over to everybody for a few last words. Aang, do you want to add anything before we go?
Well, thank you again, Sharon, for being the moderator for our panel. Again, we are grateful to the MDA. I think ultimately, as an industry person, I have to say that we are so focused on patient gene therapy drug development. And I think specific to CJ is that being a teenager and growing up with a rare disease is tough. It's already tough being 13 years old. And I understand. I mean, I was 13 and back in the day too. We're trying our best to expedite our trial to make sure that we can provide access to this gene therapy to everyone, right? And we're working on it really, really hard. And we hope that we get to work with you soon.
That's great. Dr. Johnson?
Yeah, no, just to echo those sentiments that this is a tough disease, and I'm excited to see how much progress has occurred, even just in the last few years. And as somebody who sees patients in clinic with this condition. It's been difficult over the years, and like I said, I'm hopeful that we're turning the page and really being able to see effective therapies come forward, and that myself and our consortium are ready and prepared to support these trials and ensure their successful completion.
Okay, Craig or CJ, anything you want to add?
CJ, anything you want to finish up with?
Not that I could think.
Well, I just want to thank everybody again for including us in this. We are, again, extremely hopeful that this is my wife Jody. She was on during the presentation portion. That that this is expedited in a time that it's going to be able to benefit both CJ and his brother Brody prior to getting to a point of no return, and just appreciate all the hard work that the teams put into making this a potential reality.
Well, that's fantastic. I mean, I think it truly takes a village. We've seen so much input into these studies and trials. The MDA has put in about $40 million into limb-girdle research over the years, from identifying all the genes to building mouse models to investing in gene therapy. But all of the other groups in this space, like Speak Foundation, I think it's all coming together in a way that's really supportive of these studies. And it's nice to see the discussion move beyond just Duchenne and start looking into a lot of the other forms of muscular dystrophy. So, and of course, I personally am super excited about this study. So I want to thank you all, everyone who's played a role. This has been fantastic, wonderful talks today. And thank you to SBio
Awesome.
for supporting this.
And thank you, everyone. It's a team effort. So thank you.
When somebody has a devastating disease, it affects not only the family structure, but it really affects the community at large. Disease doesn't just affect the patient, but it affects a much wider spectrum of people. SBio is an AAV gene therapy company. We provide treatments for devastating disease. Often these diseases have no viable treatment option. We have worked on Duchenne's muscular dystrophy, Pompe, hemophilia, heart failure, and Parkinson's disease. SBio was started when I gave a course at University of North Carolina. The course was teaching professors how to start spin-out companies, and in the audience was Dr. Richard Jude Samolsky. And he had a lot of intellectual property at the university. So he came up to me and he said, "I need help, but I can't pay you. And there's a lot of skepticism about this technology. But if and when we make it work, we'll change the world." Gene therapy is a very simple concept. Now the question is, how do you get the gene into the body? We use an adeno-associated virus, or AAV, as a carrier to get into the body. So what we do is we take out the wild type DNA of the AAV, and so now it's just a protein shell, and in that we can put our therapeutic payload. And for most diseases, that means a one-time treatment option for patients. Duchenne's muscular dystrophy is a neuromuscular disease that affects young boys. These little boys lose the ability to walk, usually in their early teens. Often they die by the time they're in their 30s. Today, our drug is being advanced by Pfizer into pivotal clinical trials, and we've seen the results of little boys who were treated who can barely walk 4 steps and are now in swimming lessons and Little League baseball. It took us a long time before we got our first round of financing. Today, the company is over 350 professionals operating in 5 different countries. We have 3 different drugs in the clinic, and we expect that we'll have a very strong pipeline with many more therapeutics advancing into the clinic in the very near term. I carried a note for a long time from a little boy who I met who had Duchenne's muscular dystrophy. And so he wrote me a note saying, "I doubt you're going to be able to have a therapeutic in time for me, but I'm going to look down from heaven and I'm going to be looking out for you, and you'll have a therapeutic someday for other boys." That note was really like the thing that kept me motivated. You know, we thought, and we were right, that we had the potential to treat these diseases. We just needed the opportunity to prove that we could do it. What I like most about being an entrepreneur is every day there's a world full of possibilities. You know, pick a disease, we'll work on it, and we'll make change. And that's incredible. That's really incredible.
Hi, we're the Szymczak family. John,
Denise, Dust. And are you Holly?
we live in Wisconsin and we're just outside of Milwaukee. Josh is 16, Holly is 13, and they both have limb-girdle muscular dystrophy type 2S. And at the time of their diagnosis in 2015, they were the first two ever diagnosed with this subtype in the United States. At that time, they only knew about 8 total cases in the world, and now there are close to 20, with 5, including our 2, that we know about in the United States. Our story began when the kids were born. Josh was born 5 weeks early and only weighed 3 pounds, 9 ounces. Holly came along 2.5 years later and weighed 3 pounds and 10 ounces. Both kids spent about 2 weeks in the NICU after birth, pretty much just as a precaution, but the doctors had a few concerns that they wanted to monitor them for, and they wanted to make sure that once we got them both home that they would continue to thrive. Josh's first 2 years were a little bit of a bumpy ride. We had many doctor's appointments, lots of testing. Josh wasn't meeting his milestones. He finally did meet them. He was very delayed, but his doctors hoped he would catch up by 2 years old, like most preemies do. We finally got to a point where the doctors started saying we had made it to the end of testing where they could treat or cure Josh, and we all kind of decided we need to take a break. We didn't want to know anymore. We needed a break. Once Holly was born and pretty much showed the exact same issues that Josh had, we were back at it, seeing if there was something new that we missed from when Josh got tested. In 2014, they gave us pretty much the last option, which was whole genome sequencing, which may give us an answer, but they said there was only a 25% chance of it. In May of 2015, we had our answer: limb-girdle muscular dystrophy type 2S. Once we knew, we had to face some of the challenges, and some of the greatest challenges Josh and Holly have is not being able to do all the things that their friends can do. It's kind of disappointing that they can't go swimming by themselves, hike to a waterfall when we go on vacation. They just can't get up and just go anywhere. We have to make sure we have all of their stuff: medicine, cooling vests, spare clothes, water bottles, wheelchairs, etc. before we do anything. What once was an easy task for us leaving the house now takes forever because we have to have all of our stuff.
Your stuff, right, Holly?
Yep.
Josh, we talked about greatest accomplishment. What did you say your greatest accomplishment was? It was joining the what? Joining the golf team. Joining the golf team at school. It was being able to ride your bike for how far? Bike for 3 miles. It was. And what were you in 2020? The Wisconsin what? State ambassador. Yep. Holly, what did you say your greatest accomplishment was? Was it mini golfing down a mountain this summer? Oh, and was it becoming the state ambassador? It was. And guess what? Our whole family, our greatest accomplishment was it winning a contest and having a Green Bay Packer come to our house? Yes. Who came to our house? Who was it? It was Donald Driver came to our house and had lunch with us and played video games. Yeah. It was a lot of fun, wasn't it? A memory we'll never forget. Yeah. Our whole family has really been changed a lot since we discovered the kids had limb-girdle. It makes, I guess, our family more considerate, and it's shown us that there are lots of people in the world that can use our help, even though we have difficulties with certain things. We spend much more time volunteering since the kids have come down with this. We not only devote our time and resources to MDA, we also volunteer with Make-A-Wish of Wisconsin. And as of this year, we've helped grant 6 wishes for kids in need. Limb-Girdle made you guys into more caring and considerate people that you are today, wouldn't you say? Yes. Yeah. Joshua, can you tell them what your unique fact that they don't know about you? What do you love to do? You love to bake and cook. You love to bake and cook. Is that you too? Yes. Yeah, you both love to do that? Mm-hmm. And Josh, what do you collect? Harley Davidson what? Harley Davidson pennies. No, not pennies. Poker chips. Poker chips, right. Holly, what do you love to wear on your feet? Warm fuzzies. Socks. You do love warm, fuzzy socks.
Josh and Holly would like the world to know that they're not really any different than anybody else. Although they may not move as fast as others and have trouble getting around, they are just like everyone else. Josh likes to remind us that him and Holly are just like chocolate chip cookies, but without the chocolate chips. They're still cookies, they're still delicious, just a little bit different.
I guess I would say the one thing we wish we knew earlier in our journey with Limb-Girdle is probably that there are a lot more resources out there that we didn't know about at the time. There's lots of programs available and different things that we slowly learned about as we went along, but if there was a way to make it so it was available right away, it would be a lot easier, I guess. We wouldn't have struggled as much, I would say, wouldn't you?
Yes.
If Limb-Girdle was cured today, what do you say you want to do, Josh?
Running would be fast.
Right, and you want to win a running race, right? Holly, what did you say you wanted to do? Go hiking with us?
Yep.
And do you want to hike without having to hold my hand and go by yourself?
Yes.
Thank you all for listening.
Thank you.
Thank you.
Hi, I'm Kat Bryant-Knutson. I wanted to share with you that I was diagnosed with LGMD type 2I in 2006. At that time, there were very few resources for Limb-Girdle. So in 2008, I founded the Speak Foundation to specifically help those living with muscular dystrophy. The one thing I knew is that we needed a way to communicate to everyone in this vast community. So we started the LGMD Patient Network. It is the first patient-led registry for all forms of limb-girdle muscular dystrophy. The LGMD Network is for any individual to join. And when you join, you receive a free subscription to the world's only LGMD news magazine. The magazine brings together researchers, clinicians, and pharmaceutical companies all in one place. We've teamed up to give you the best information that is the latest news for the LGMDs. If you haven't joined our network, please sign up at thespeakfoundation.com. Thank you.
Hi, I'm Andrew Robertson. I'm 40 years old and I'm from Canterbury in the United Kingdom. This is a very brief overview of my LGMD journey. I was diagnosed with muscular dystrophy at the age of 19, and I was very fortunate that it was a very quick process to diagnosis. Some people go for years not knowing something's wrong, but not knowing what it is. I was seen by a medical professional who recognized it straight away. And the path to diagnosis was very quick with the genetic testing. I was told three things though. I was told the condition isn't treatable. There's no cure for it, and the best I need to do in my life is to concentrate on quality of life. And when you're 19 years old and you're told those three things, and you know that the prognosis is a wheelchair one day and is giving up walking, it can be crushing. It can be very difficult to regroup. But I held on to this kind of this belief that I would continue walking. I'd do everything possible, stay as fit and healthy as possible, pursued a drug trial in 2006, part of the YF trial of a myostatin inhibitor, which was pretty good until the trial was halted. And one of the images you can see there is me in an exoskeleton walking suit, not giving up on the dream of walking again. Technology has helped me do a bit of a robot dance. But over the years, I had probably 10 years of walking before I used a power chair. And that first moment sat in the power chair and realizing that I could go out, it was very liberating. I wasn't scared anymore. I could zoom around town without the fear of falling over. If there was a windy day, it's not a problem for me. If it's snow and ice, a little bit of a problem, but not as much as when I was walking. So what I feared the most actually turned out to be what gives me the most freedom. I live without pain, I live without fatigue, and my little chap, who's now 8 years old, will still ride on the chair with me. He still thinks it's fun. And in fact, today he said to me, "Dad, being in a wheelchair, it looks fun. Is it fun?" And you don't want to crush him and say, well, it's not always fun. But it's making the most out of life. It's saying that you've been dealt a very difficult hand, and unless you can take that by the horns and say, you know what, I'm going to travel, I'm going to go places with the help of some wonderful people. And in the bottom left, I've got some of the caregivers that have helped me to travel, and my motivation, which is my son. He wants to go everywhere and see everything, and I'm really excited about doing that with him. Things are possible, and traveling can have its complexities, but with the right research, especially when you look at online forums and Facebook groups and people that are very, very experienced, have a lot of knowledge to impart, learn from them. I did, and it gave me the confidence to go out there. And sometimes planning is key, but confidence is also key as well, because you can plan to the nth degree, but unless you're prepared to hold your head up high, go out there, smile, and make things happen, life's not going to happen without that happening. You don't want to let things pass you by. I find a lot of positivity has come out of this condition for myself, meeting a lot of wonderful people. Most of the time people smile and I smile back. Doors literally open for me. And I think that's something that I'd perhaps not really realized. When I was younger, I always had a very strange belief that I would be in a wheelchair one day. And I always said if I could enjoy a nice sunny day in the countryside, getting back to nature, then I'd be okay. And it's very strange how that then turned out to be a reality. I have a very good all-terrain wheelchair which allows me to go outside and enjoy nature, especially with my son. Having all the right equipment really does make life that much easier. I'm now 40 years old and I'm still holding on to that belief that one day I'll be walking again. The next treatment that might be on the horizon, the gene therapy, the holy grail for anyone with LGMD, could be there for me. And I do hold on to that positivity. I don't rely on it anymore. I'm happy without it. I'm extremely content with what I am. But a little part of me will always look to the future and hope and pray that it's got a different ending for myself and other people like me with LGMD.
Diagnosing LGMDs is very challenging. Many of us in the patient community have stories of long diagnostic odysseys and often misdiagnosis. In this session, moderated by Laura Rufibach of the Jain Foundation, clinicians and geneticists will go over the diagnostic process and explain why it can sometimes be difficult. Also, a group of patients will share their real-life examples of their diagnostic journeys.
Thank you, Brad, and welcome to session 7, entitled Diagnostic Landscape for LGMDs. My name is Laura Rufibach, and I'm the co-president of the Jain Foundation, whose focus to identify potential treatments for LGMDR2, also referred to as LGMD2B or dysferlinopathy. Before we get into the session, I'd like you to consider what it means to have a diagnosis. In my opinion, a diagnosis is made up of 3 aspects. The first is a clinical diagnosis, which is the process of identifying a disease based on the examination of the symptoms, such as which muscles are weak. For the LGMDs, this can usually only get you so far with your diagnosis because the various LGMDs subtypes have similar and overlapping symptoms, so it is very difficult to distinguish one subtype from another based solely on the clinical symptoms. For example, your doctor can differentiate between an LGMD and a distal myopathy because different muscles are weak. The proximal muscles like your hips or upper legs in the LGMDs, and your distal muscles like your calf or lower leg in a distal myopathy. But they can't differentiate between the different LGMD subtypes because they all show proximal weakness. Therefore, further testing is generally needed in order to identify the specific LGMD subtype that you have. The second aspect is molecular testing. This includes further testing such as blood tests, imaging tests, biopsies, and protein evaluation. This molecular testing can provide information that could direct your physician towards a specific subtype. For example, if you have absent dysferlin protein based on evaluation of your muscle biopsy, that is highly indicative of a diagnosis of LGMDR2. For genetic-based diseases like the LGMDs, the final level is genetic testing, which involves the evaluation of your DNA to identify the disease-causing variant or variants. For the dominant LGMDs, you need 1 pathogenic mutation, and for the recessive LGMDs, you need 2 pathogenic mutations in the same gene in order to have a definitive genetic diagnosis. Unfortunately, despite the power of genetic sequencing, it is not always possible at the moment to get a confirmed genetic diagnosis because of the identification of variants of unknown significance, or the gene responsible for the specific type of LGMD has not been identified yet. However, new advances in genetics are continuing to be made and new genes identified so that those without a confirmed genetic diagnosis today may be able to get one in the future. Lastly, I would like to have you keep a few important things in mind as you listen to these talks. First, knowing the underlying cause of your muscle disease, whether it be mutations in a specific gene, neurological issue, or an autoimmune disease, is very important to understand that as it affects your disease management, progression, outlook, treatment options, and family planning. Therefore, it is important to continue testing until you have a definitive answer to what is causing your muscle disease. Second, a diagnosis is not based on just a single test, even for genetic-based diseases like the LGMDs. It is the combination of the results from all the testing done, clinical symptoms, molecular testing, and genetic testing, that your physician analyzes together to come to your diagnosis. Therefore, don't just rely on only one aspect of the diagnostic process that I described, but look at the results from each part together to see if they match and are telling the same story when making a decision regarding your diagnosis. The three expert speakers in this session will touch on all these things in their presentations today and will provide you with information about whether there are additional things that you and your doctor can do to help you get a more definitive diagnosis. There will also be several individuals that will share their real-life diagnostic journey. Our first speaker is Dr. Jorge Bevilacqua, an MD neurologist and PhD researcher from the University of Chile in Santiago, Chile.
Hello, thanks Laura Rufibach for the kind invitation and the organizers of this Congress for inviting me to give this presentation. We will talk about some concepts, the definition, the classification, and some other aspects of Limb-Girdle muscular dystrophies across the world. This is my conflict of interest statement and the outline of the presentation. So as I said, we will discuss briefly what we understand for a limb-girdle muscular dystrophy, how we reach to the current classification, how is it distributed across the world, and discuss briefly if there are other diseases that look like limb-girdle muscular dystrophies. That's very important for you to know. And what tests and what's the main strategy we use to reach the diagnosis. And before talking about limb-girdle muscular dystrophies, for me it's very important to make the point of what we understand for limb-girdle muscular weaknesses, which is not exactly the same. For weakness, we mean the impairment of the motor function of both the shoulder girdle muscle or the pelvic girdle muscles that could present either together or individually in some patients, but that's a very unspecific and descriptive term and a clinical definition, what we doctors call a syndrome, right? So a number of signs and symptoms that the patient presents. That could be genetic or acquired. That could be due to different diseases, for example, motor neuron disease, like in spinal muscular atrophy, or given the situation of the neuromuscular junction in the congenital myasthenic syndromes, we have some patients that they present with limb-girdle muscular weakness. And of course, it could be due also to muscle disease, as in myopathies, but myopathies in general could be hereditary, toxic, inflammatory, infectious, and they all give this, or may give this presentation as a limb-girdle muscular weakness. So then when we say that is a limb-girdle muscular dystrophy, well, when this weakness is coming from a genetic disease that there is presenting clinical, imaging, and histopathological findings of muscular dystrophy. So it's a primary disease of the muscle that has to be defined as a dystrophy. We will go briefly back to this concept later on. So many muscular diseases can look like a limb-girdle muscular dystrophy. As I said, that phenotype could be caused by different myopathies and genes, not only the ones that produce a limb-girdle muscular dystrophy. It could be dominant, recessive, or X-linked. It could appear also in inflammatory myopathies, but by definition, we have agreed, I mean, we, the scientific community has agreed that a limb-girdle muscular dystrophy will never be a dystrophinopathy, a facioscapulohumeral muscular dystrophy, a myotonic dystrophy, or a distal dystrophy or distal myopathy. So those phenotypes and diseases are ruled out from the classification of a limb-girdle muscular dystrophy. Here is a table from this paper in which some genes and protein products and all nomenclature of the limb-girdle muscular dystrophies, diseases from this list are with a limb-girdle muscular phenotype, but they are not classified as such. So it's very important for you to retain the idea that many different diseases and genes could cause a limb-girdle muscular dystrophy phenotype, but will not be classified as such. And where that concept of limb-girdle muscular dystrophy comes from, it comes from the work of Lord Walton and Professor Natras in the 50s, where they classify about 100 of the patients with myopathy, and they find a group that were more or less alike with this kind of distribution of the weakness, and they were late in comparison in the onset. They were late in comparison, for example, with other diseases like Duchenne muscular dystrophy, and that the predominant weakness was in the girdles that were usually autosomic or autosomal recessive, and they were of a relative slow course of the disease. That was a concept made according to the knowledge and the scientific knowledge of the time, but later on this concept was quite discussed and controversial. Another person that contributed much to the myology from England, Newcastle, he said in the 70s that it was very difficult to understand this concept because it was more than a disease, it was a collection of different things put together because they share this common phenotype of the weakness of the hips and the shoulders. And he pointed out, visionary, that there were a variety of the patterns of inheritance, onset, progression, and distribution that required to redefine what we understand for a limb-girdle muscular dystrophy. In those lines, in 1995, 20 years later, the first European Neuromuscular Center workshop was gathered to discuss about the, at the moment, the new genes and new loci that were going to be identified to put them in order and to classify them correctly in order to have an understandable classification of limb-girdle muscular dystrophies. At that meeting, it was established that the limb-girdles were going to be classified as type 1 when they were dominant in inheritance, or type 2 when they were going to be recessive, and a letter of the alphabet was going to be put just to indicate the chronology of the discovery of the loci or the causal gene. For example, in the case of limb-girdle muscular dystrophy 2A, calpainopathy, the 2 refers that it was recessive, the A because it was the first identified gene causing this form of limb-girdle muscular dystrophy. So in the following years, that list was filled up with many different types of limb-girdle muscular dystrophies, even reaching quickly to the end of the alphabet. So that was becoming a problem for two main reasons. First, because we don't have more letters on the alphabet, so the classification was going to be very difficult to understand. And second, because even though the definition was more or less established, some categories of disease were included as limb-girdle muscular dystrophies, but they were not fulfilling totally or completely the concept of limb-girdle muscular dystrophy. This classification is important still to retain, because we are still using it, and most of the diagnoses you have received are made based on this nomenclature and classification. So, because that was difficult to understand these, the order of the diseases with that classification, in 2017, a new European Neuromuscular Center workshop was gathered just to review the classification and reach a consensus on a new classification and the definition of what a limb-girdle muscular dystrophy is, and to try to make a new classification that will be useful for research, for understanding, for the new treatment trials that we're going to come. In this new classification from 2018, published in 2018, the limb-girdle muscular dystrophy were classified as D for dominant or recessive, and instead of putting a letter to make the order of the type, it was a number was put. So in a way that dominance will be 1, 2, 3, 4, 5, etc., and recessive alike, and the protein product or the gene affected should be mentioned explicitly in the name of the category. So, for example, for calpainopathy, limb-girdle muscular dystrophy, R for recessive, 1 because it's the most frequent. This concept of the frequency is something that probably will need to be reviewed, because we are discovering that the relative frequency of the different forms of limb-girdle muscular dystrophy is not exactly what we thought it was before. So for the time being, that's the new form of naming the limb-girdle muscular dystrophies. The other main point coming from that meeting is the current definition. A limb-girdle muscular dystrophy is a genetically inherited condition that primarily affects skeletal muscle, that is progressive and dominant on the limb girdles, let's say the shoulders or the pelvic girdle, as in this woman, that the patient needs to achieve independent walking before the diagnosis is made. This is to separate the limb girdles from other more early presenting dystrophies, that there should be at least 2 unrelated families described with the condition, because some entries in the old classifications were based on just 1 patient described across all the whole wall. Elevated serum CPK, as always, degenerative changes on the MRI, just to rule out, for example, inflammatory myopathies where there are changes in the MRI, but they resolve later with treatment. In dystrophies, those changes are terminal and degenerative, and they will not go back to normal unless, hopefully, in the future we could treat them effectively. And that in the biopsy, there should be dystrophic changes. This is quite complicated for this audience, but at the microscope, not everything that is abnormal in the muscle is a dystrophy. So with that definition, the old classification was reviewed. Some categories were put away from the classification. For example, here in the dominance, 3 forms, the 3 first forms of the old classifications were eliminated because different reasons, and a new classification considering the new definition and considering some forms of limb-girdle muscular dystrophy that fit the definition but were never included in the classification were now added, as in the case of collagen 6 related myopathies. Similarly, for the recessive category, we have a long list of diseases. The first 15 are more or less the same than in the old classification, but some changes occur in the second half of that long list, with some diseases that were removed from the old classification and some others that were added, just fulfilling the current definition. It's important for you to know that all these symbols and names and proteins that we mentioned and we use to classify those diseases are very important functional or structural components of the muscle fiber or their immediate environment. So those are molecules that are very, very substantial for the function of the muscle. And if any of those is altered or deleted or removed from the muscle fiber, the patient will develop a dystrophy, as you know, or a muscular disease. What about the types of limb-girdle muscular dystrophies across the world? In 2014, this was more or less the map of the prevalence of these forms of disease. As you can see, the information is the best patchy, with some information from North America or Brazil in this case, some other from Europe. And as you can see here in the color code map, some forms of limb-girdle muscular dystrophy were more prevalent in some parts of the world, as for example, limb-girdle muscular dystrophy 2I that was described mainly in the north of Europe, same with anoctamine for Finland and things like this. And so this map is telling us two things. First, that limb-girdle muscular dystrophies are present all over the world, but that the types of the limb-girdles are not, in numbers, are not the same in the different regions. So what we need to know is that depending on when you are, the frequency of the forms of limb-girdle muscular dystrophies could vary substantially. With the advancement of all the next generation sequencing strategies, some studies started to analyze large numbers of patients with limb-girdle muscular weakness and to try to establish what were the genes more frequently affected in the different regions of the world. There are many works coming up and there will be much more in the future, but at the moment there are three main projects that gave us very interesting information. The first one I would like to mention is this one that is including countries from Europe, North Africa, Middle East, the MAIOSEC experience. And in this study, 429 genes were analyzed at the complete exome, genes that are obviously related to neuromuscular disorders. And they reach the diagnosis in about a half of the patients with this strategy. Interestingly, about 8 to 10 genes, most of them included in the new classifications of limb-girdle muscular dystrophy, justify half of the patient's diagnosis. It's important for you to know that even with this very good approach, the yield of the diagnosis is about a half. It's not that you will be able to reach the diagnosis in 100% of your cohort always. It still need to be reanalyzed. This, the other important information that is coming from those studies is that this form, the calpainopathy and the dysferlinopathy, are the most frequently found limb-girdle muscular dystrophies genes affected. In another approach, quite similar, a study from North America, the United States, with a 35 gene panel, the diagnosis was reached in about 27% of the patients, so let's say about 30%, 1,259 cases. And again, the most frequently found limb-girdle muscular dystrophies were calpainopathy and dysferlinopathy, followed with a different proportion for other forms, mainly the fukutin-related protein form and the ANO5. But as you can see here and in the previous study, and in this one from Latin America with a smaller panel of 10 genes only, in which diagnosis was reaching only 16% of the patients, 300 and something. Again, dysphalinopathy and calpainopathy are the two most frequently found limb-girdle muscular dystrophies. So even though there are regional differences in different parts of the world, it seems that globally, the relative frequency of the genes affected and that are producing limb-girdle muscular dystrophy are similar across the entire world. Another important concept that I would like to mark for you to know is that not, as I said before, not all the limb-girdle muscular dystrophy phenotypes are produced by genetic disease or are produced by mutations in neuromuscular genes. This is an example of a 4-year-old girl with a subacute limb-girdle muscular weakness that it was initially diagnosed as a limb-girdle muscular dystrophy because she, strictly speaking, fulfilled the definition that we use now for limb-girdle muscular dystrophy. The CK was quite elevated. There was a dystrophic part in the biopsy, and there was no particular labeling of with the immunohistochemistry. I mean, there were no proteins missing on the biopsy, and the muscle MRI showed diffuse fatty replacement, which, as I said, fulfilled the definition of limb-girdle muscular dystrophy. But the problem came when we did the next generation sequencing study, including all the genes for limb-girdle muscular dystrophy known, and there were no mutation found. So at that point, in a routine control, one of the colleagues saw that the little girl was improving spontaneously. So that prompted us to consider that the diagnosis could not be a genetic myopathy, but an acquired one. And we reviewed the algorithm of the study of the girl, and some part of it was missing. And what we discovered is that there were an increase in the anti-HM-GCR antibodies, a type of antibody that could produce a phenotype that is remarkably similar to a limb-girdle muscular dystrophy, but constitutes a treatable disease and is not a dystrophy, it's not genetic, it's an acquired one. So the point here is that even when the medic or the patient is, the suspicion is of a limb-girdle muscular dystrophy, some check-ins should be done to exclude other forms of acquired myopathy. So in line with this, the question comes is how we reach the diagnosis of a limb-girdle muscular dystrophy. Traditionally, what we do, what medics do, is to have a good clinical history and assessment, examination of the patient. And when we have an hypothesis, we go through a list of different ancillary tests that help us to demonstrate that our hypothesis is correct. There are some tests that are quite accessible and are considered routine, like, for example, the lab biochemistry, the levels of CK, and some others are chosen depending on the hypothesis and the resources of the place where you are working on. Traditionally, as I said, we went through all this more or less in that order and reaching the possibility of studying molecular genetics at the end of the list once we define more or less which genes were the potential causing ones for the patient's ailment. This is what we used to do because, as I'm going to tell you in the next slide, things are changing a little bit. It's also important to know that this is quite variable and different in different parts of the world, given the different kind of resources, the accessibility, and also because the different uses that the medical community may have in one or another place. So on those lines, what classically was done before is, as I said in the previous slide, that when a patient was being suspected of having a neuromuscular disorder, the medics assessed the patient and started to study with some electrophysiology and imaging and biochemistry, and then the following step usually was to take a biopsy to make all the analysis, and including immunohistochemistry or Western blotting, in the case it was accessible, to choose a single gene or a few genes as a candidate, and then the screening of those genes was done one by one. That was very expensive, that was very laborious, and in the better hands, you could have a yield, a diagnostic yield of about a quarter of the patients through this methodology. With the next generation sequencing, accessibility, and lowering of the costs, what we have changed is that we still assess the patient clinically and with some routine laboratory. But instead of doing all this list of things that sometimes are very expensive, very invasive, and sometimes they don't exist in some parts of the world, we now may jump directly to doing some genetic testing through panels that are much better cost effectively. What I'm trying to say is that in just one test, we can analyze more than 100 diseases according to our hypothesis, and that will cost no more sometimes than a few cents of a dollar for each gene studied. With this approach, as I showed in briefly in some of the studies we commented before, we may be having diagnosis in about half of the patients without the need of going through those expensive and invasive assessments. So if you can reach the diagnosis this way, which happens in about half of the patients, the diagnosis is done and the effort will be concentrated in help the patient to deal with the ailment they may have. Sometimes, however, even having all the study and having the genetic analysis, we may find a situation that the complete study algorithm gives no answer and no diagnosis, or even worse, after the genetic analysis, we may encounter this temerary variants of unknown significance, which is a nightmare for many patients and doctors. What should we do if that's the situation? And that situation will happen very frequently in the future. What we should do is what we always have to do, is that review all the patient phenotype, the clinics, all the ancillary testing, and reconsider whether there was something missing, or if the patient has been tested with the appropriate test. Because, for example, as in the case I showed you before, a patient that is analyzed for a genetic cause of the disease will never appear as a positive one if the patient is having an autoimmune disease. So the idea here, in simple, is that always the phenotype and the clinical manifestations of the patients should be rechecked to see whether we are doing a correct job there and it need to be amended in some way. So to finish, some take-home messages to retain. Definition of a limb-girdle muscular dystrophy is quite unspecific and is constantly changing. There is not just one disease, it's a group of diseases that could be dominantly or recessively inherited and are classified as dystrophic myopathies. Remember that dystrophic for the medics mean something quite sharply defined. It's not just any myopathy. The frequency of the different forms varies across the world with regional particularities. So it's important for you to know that what kind of diseases are more prevalent in your country, in your environment, just to go for those first in the diagnostic algorithm. In general terms, limb-girdle recessive 1 calpain related and 2 dystrophin related are the most frequent forms across the world. And two more things. Remember always that many diseases look like limb-girdle muscular dystrophy. So the medics and patients should have the idea that in front of that manifestation, it should be check which diagnosis is precisely the one that is affecting a particular patient, that we need to rule out some diseases that are not genetic, but that could mimic a limb-girdle muscular dystrophy. And in general, my recommendation will be that we do the genetic testing first through those not invasive panels, which are cheaper nowadays, before going to invasive and expensive tests. And finally, you need to know your limb-girdle, because that, in the first place, will help the medical community to understand the nature of the disease is affecting you. You need to have an adequate genetic counseling according to which disease or disease subtype you are having, to prevent the known complications and morbidities associated with your condition. And finally, and most important, because it's the only way you will be able to access to trials and emerging treatments that, as far as we know, will be tailored not only to the gene and the molecule affected, but sometimes tailored to a particular mutation that is affecting patients. So with this, I finish.
Hi, thanks for the opportunity to participate at the LGMD conference. My name is Eleonora, or Noni as my friends call me. I'm originally from Bogota, Colombia, and we are 4 siblings in my family. I am the youngest. My sister and I both have LGMD, but I was the first one that was diagnosed. My 2 brothers and my parents are healthy. I was diagnosed when I was 12 years old, and the earliest symptom that I can remember having is when I was about 5 or 6 years old. I started walking on my toes, and my grandmother was scolding me because I was walking as if I had pooped my pants. So I remember that vividly. But looking back, I also remember that I couldn't run as fast as other kids in my class, or jump rope, or play sports as well. But regardless, I was a tomboy, and I could climb trees and ride a bicycle, skateboard, and try to do everything that my brothers did. When I was about 9 or 10, I began having other issues like falling down frequently, having trouble getting up from the floor or walking upstairs. So my parents started taking me to different doctors who couldn't figure out what was wrong. And finally, an orthopedic doctor thought that I might have a neuromuscular disease and told my parents I would not be able to get a proper diagnosis in Colombia at that time, so they should try to bring me to the US. In 1979, my parents brought me to Massachusetts General Hospital in Boston, where they did all the routine tests available at that time. I had function tests, blood work, x-rays, an electromyogram, and finally a biopsy of my thigh, which left a big scar. All these tests revealed muscle weakness in my hips and shoulder area, a CPK of over 2,500 lordosis, contraction of the Achilles tendons, among other issues that were typical of LGMD. And I was examined by this wonderful and caring doctor, Dr. Krishnamurthy, who diagnosed me correctly with limb-girdle muscular dystrophy. In 1979, there were no subtypes or genetic testing, so this was the final diagnosis. When I was 17, I started having some foot drop issues, which made me trip frequently. So my parents took me to see a doctor at Miami Children's Hospital who recommended AFOs. And while I was waiting for the doctor to finish taking the measurements, another doctor who was an MD expert came into the room and started telling me stories, unsolicited, about surgeries that he had done on other patients with muscular dystrophy. And he told me, quote unquote, not to worry, that when I could no longer hold my head upright, he would transplant a muscle from my calf onto my neck so that I could hold my head again. I didn't have any problems with my neck then, and I don't have any problems with my neck now. So I was very traumatized by his stories, and I did not want to become anybody's guinea pig. So I refused to see another muscular dystrophy doctor ever again. Several years later, my sister, who is 7 years older than me, was also diagnosed with LGMD. Like me, she started walking on her toes when she was about 10 years old, but she was enrolled in ballet lessons and had casts on both her legs to stretch the Achilles tendons. Her disease progressed much slower, and although she wasn't very athletic, nobody really thought that she had LGMD. The onset of her disease started when she was 25, and she began going to the gym and lifting weights to try to get more fit. However, her muscles would burn and were getting weaker instead of stronger. So she went to the hospital where they performed tests and diagnosed her with LGMD as well. For several years, I had been reading online about the subtypes and inquiring about it here and there, but it wasn't until 2014, thanks to my sister, that I joined the Facebook group LGMD Beyond Labels and Limitations and started meeting all these people with so much knowledge about the disease. And among them was Brad Williams, who also has LGMD and who happened to have studied at the same university and during the same period of time when I was there. And yet we ended up meeting for the first time in this group. When I joined this Facebook group, I didn't know my LGMD subtype, and I had been told before that I needed a muscle tissue to find out, and I didn't want another biopsy, so I hadn't inquired anymore. But thanks to the wonderful people in this group, I was made aware that there was a consortium of LGMD foundations that was sponsoring a free genetic testing program using next generation sequencing. And through this program, I was able to send a saliva sample in the mail. And 4 months later, I got the genetic confirmation of my LGMD, and finally the diagnosis of subtype 2A, or R1 as it is known now. In summary, my initial diagnosis was in 1979 and the final diagnosis in 2015. So it took 36 years to get the genetic confirmation of the LGMD and to find my subtype. If I started having symptoms today, I would probably be diagnosed much sooner and faster thanks to the genetic MD panels and other knowledge that has come about with research. I think genetic panels are a wonderful tool that may avoid a painful biopsy for many people, and they would be very important to have free genetic testing in other countries to be able to get a proper diagnosis without having to travel to the United States. And thank you very much for listening to my diagnosis story today.
Hi, I'm Andrea Klein, and my story really begins in infancy. My mom had noticed that just like my middle sister Cheryl, as a baby, I couldn't crawl. I scooted on my bottom and I tucked one leg inward, put one leg outward, and sort of rocked side to side to propel myself. Also, like my sister Cheryl, who was 5 years older, my neck began to lean to one side. Eventually, this was diagnosed as torticollis, a weakening of a muscle in my neck, but they thought this was a separate birth defect from the other symptoms that we were having, like challenges getting up from the floor, climbing stairs, and running, and really keeping up with our peers. Over time, eventually, we arrived at our initial diagnosis, which was really done from my sister having a muscle biopsy. And of course, she had the whole needle test, that awful part as well. But they decided, because I had had a recent torticollis correction surgery, that I wouldn't have the muscle biopsy. And because our symptoms were the same, that we would go based off of her test results, and that would also be my diagnosis. So that was late 1980. And at that time, we were told it was limb-girdle muscular dystrophy. Now, flash forward many years later, when I was 33, in 2007, my middle sister Cheryl passed away from respiratory failure. Up until that point in time, I had never really questioned what my diagnosis was. But because we were not told to expect respiratory involvement, we were instead told to expect cardiac involvement, and loss of ambulation, that really made me want to find out exactly what condition that we had. And it wasn't much longer after that that I was able to go online and search and learn about limb-girdle subtypes. Now, I was, of course, trying to find a subtype that referenced pulmonary function decline. And the only one that I could really find was Limb-Girdle 2I. And eventually I was led to a Facebook support group for individuals with 2I. It was through that group that I met someone from a nonprofit for 2I that was able to help me get a free genetic test for that disease. Unfortunately, my test results came back negative, so that left me with more questions than answers. Over time, I met other individuals with limb-girdle, and I kept coming back to that, but I just couldn't find a subtype that fit. Well, eventually I attended a Speak Foundation conference in 2013, and it was at that conference that I had agreed to participate in a presentation with Brad Williams, and he was presenting a tool called ALDA or ALDA. And through the course of that presentation, I answered questions that would help to identify the probability of a limb-girdle subtype. And so the result of that was a 97% probability for something called Bethlem myopathy. Now, the odd thing about this was also at that conference was a researcher who was very familiar with that disease. And he had actually sat at my table for lunch that day, and I was unaware that he had noticed several characteristics about of the disease in me. And during Brad's presentation, he asked if he could get up and come and look at my hands. So he examined my hands and pointed out, again, some distinct characteristics of a collagen 6 myopathy that I had. And that really was extremely shocking to me, and I was anxious to rush back to the hotel room and read more about that condition. Ultimately, what happened was I finally, after 5 years of not seeing a neurologist, saw a neurologist and shared those findings from that ALDA tool with him. And much to my disappointment, he looked at the literature that I shared and disagreed. And while I was there, he insisted that I be tested genetically for Pompe disease. Well, of course, a few weeks later, those test results were negative too. But he was so convinced that I had Pompe disease that he ended up sending me a test kit that I was able to have done locally to get a second genetic test. Well, of course, that was negative as well. So at that point, he didn't really seem to have any other recommendations, and I kind of felt hopeless about the whole situation. I would say several months later, Brad had reached out to me because he had found online a National Institutes of Health clinical diagnostic study that was led by Dr. Karsten Bonneman. And he shared the information with me. I reached out to them. I had to get copies of my records from my neurologist. I had to get a CK level test. And eventually I was accepted into that study, got a huge packet of papers that I had to fill out. And in mid-October of 2014 was when I was scheduled for a 2-day visit at the NIH in Maryland. I had been there for probably only 30 minutes. Dr. Bonneman had performed part of my exam, and he stopped and he said, "Well, I would like to welcome you to the Collagen 6 Myopathy Club. He said, it looks to me that you have all of the distinct features, but at this point we need to continue with your 2 days of testing, your genetic test, your skin biopsy, muscle MRI, and it was a few other things. And let's get those test results back just to confirm it. And of course, it was a couple of months later, towards the end of December, when most of those test results were back. And the first part of January of 2015 was when I got the phone call from the genetic counselor to confirm that I did in fact have a collagen 6 myopathy. I was intermediate on the spectrum between Bethlem myopathy and Ulrich congenital muscular dystrophy. And I guess it was in 2017 that a Limb-Girdle subtype for this autosomal recessive condition was identified as R22. So started out in 1980 being told I had Limb-Girdle, and then things sort of came full circle to learn that a Limb-Girdle R22 was on the collagen 6 myopathy spectrum.
Our next speaker is Dr. Chris Weihl, an MD neurologist from Washington University School of Medicine in St. Louis, Missouri. Over to you, Dr. Weihl.
Great. Thank you, Laura, for that introduction. And I guess I'll get started with my talk. One of the things I wanted to primarily address today is the idea that patients often are in diagnostic dilemmas, and clinicians are also in diagnostic limbo as well. And part of this relates to variants of unknown significance. And so the title of my talk is Variants of Unknown Significance. What do they mean? And I hope to let everyone fully understand what it means whenever a genetic test result has that, and how that relates to us understanding diagnosis. I want to bring up one point, and I've been trying to do this in all of my talks, is to really elucidate even how the inclusion of Indigenous people and underrepresented minorities is important in all aspects of research, just not clinical trial development, but even in genetics. And in order for us to really understand variants of unknown significance, it's important that we have genetic data not just on individuals with disease, but also individuals without disease, and that these individuals without disease are from a broad background. And so one of the programs the NIH has is called All of Us, and I encourage you to look into it, but it's a way of broadening much of the genetic databases so that it's inclusive not just of Europeans and US citizens that are predominantly white, but also those of underrepresented minorities and Indigenous people. So I'll start with just kind of the framework of genetics, which is really that we have a mutation or a gene, I guess I should say, in DNA. So this represents a gene in DNA. So DNA is a double helix. It's the one copy of one of these strands is from your mom, and the other copy comes from your father. And this gene is transcribed into an RNA, and that RNA is then the recipe that then gets created into a protein through a process called translation. And just so that we're all kind of understanding on the same level, we can think of DNA as being kind of the cookbook, and each one of these tabs represents a gene or a recipe. And just as you might have this cookbook that your grandmother may have, and she then sends out recipes, she doesn't just give one recipe out, she gives many recipes out to many different people in the family, and that's how RNA is. It's transcribed multiple times. And that allows us then to make the recipe, in this case, a cake. And sometimes what happens is we get a variant or a mutation in this recipe that makes us make a different type of cake. And I want to point this out because we didn't not make a cake. We still made a cake, but in this case, the cake looks a little bit different than the previous cake. What I don't know from looking at this cake is, does it taste the same? Is it really, can we really still call it a cake? And so that's some of the areas that we think of with variants that happen in disease genes. And so I bring up again this recipe here, and the way we think about genetics is I can have mutations that are created in this recipe. And so you can imagine this recipe here. You can imagine that we deleted all of the sugar and all of the cocoa, and then we did the recipe. And you and I pretty much know that that's not going to be a cake. That's going to be a pretty terrible cake. And so that's where I'm pretty confident that I could interpret that that cake is not going to be the right cake. However, what if this part's deleted? What if I forget to preheat the oven? I just turned it on to 145 degrees. I'm probably still going to get a cake, and it may taste almost the same. And so that's a mutation that's happening in this recipe that is a variant that might be tolerated. Another example is, well, maybe there's a mutation in this recipe that gets rid of buttermilk and just leaves milk. I'll probably get a cake. It may taste different, but I guess that's my point, is that sometimes I don't know. What if I baked this cake for 120 minutes? I don't actually know. I might still get a cake. I might still get a pretty good tasting cake. And so, or I might get something that's burnt. And so the point is that we're trying to make predictions in the recipe and trying to understand whether that prediction or that variant is going to cause a problem in the protein being, in this case, a cake. And so whenever someone gets genetic test results, often we get mutations in the recipe. And so in this case, in this gene here, AMPD1, there are a mutation or a protein change that's occurring in that gene. There's other genes as well. And how do I know that this mutation here didn't just change the flavor, but I'm still getting the protein? Or how did I know that this one here isn't completely causing a non-functional protein? And I can't tell that just from looking at genetic data. And so that's the framework with which I'd like to think about the genetics and variants of unknown significance. And so the goals of what I want to talk about are types of genetic testing, barriers to genetic testing, understanding what I just said, which is genetic variation, variations that happen in the recipe, and then how do we interpret those variations and interpret genetic test results. And so genetic testing performed in clinical practice is done in a couple of different ways. One is individual gene testing, and that can be appropriate if a family member is already diagnosed with a form of limb-girdle muscular dystrophy and they know that they have a mutation, and that would be an appropriate way to do that test. Typically, what we do are panel-based gene testing, and that's genetic testing for all the known or likely limb-girdle muscular dystrophies. There are different panels offered from different genetic testing companies. So if someone says that you had panel-based testing, your panel could be different than somebody else's panel. In addition, depending on when your genetic testing was done, older testing panels have fewer genes than newer panels. And so it may not be inappropriate if your doctor says, I don't know what type of muscular dystrophy you have, to repeat a gene panel, because the gene panels have expanded and they've identified more and more variants in different genes. Another approach is whole exome sequencing. And this is an approach that sequences every known gene in somebody's DNA. And that can be appropriate as well, but part of the problem with doing a whole exome sequence is that you're going to identify mutations in genes that are unrelated to limb-girdle muscular dystrophy, or at least known causes of limb-girdle muscular dystrophy. And that can actually add more confusion sometimes, in which case panel testing may be appropriate for first line. And finally, there's something called whole genome sequencing, and that sequences the entire genome. That's something that can be done as well. It can be challenging to interpret. Part of the reason it's challenging to interpret is because it is so much data, and it can be often even hard to handle that data. So when I think of genetic testing, I think often about some of the barriers to genetic testing and diagnosis. And some of these barriers are in our control and some of them are out of our control. And so one of the barriers is payers. So will a insurance company pay for genetic testing? This has been actually almost abolished because of free genetic testing programs. And free genetic testing programs have really taken that barrier away. Another barrier is patient knowledge. Patients don't realize that they might need genetic testing. They might have been told they've got a form of limb-girdle muscular dystrophy, but they've never had genetic diagnosis performed. And so that's another where patients and even patients' family members need to be educated on the importance of genetic testing. You might say, how is that a barrier? Well, if I can't get a patient to get genetic testing done, then I can't do genetic testing. So that can be a barrier. Community knowledge, and this conference is great, and this opportunity to talk to you is great because I can educate the community that making sure you have accurate and recent genetic test results. I advocate that patients have their genetic test filed away, just like they have their Social Security card filed away, so that they know exactly what their genetic mutation is. Physician knowledge. Not all physicians know about genetic testing and what needs to be done and whether a different panel is different than another panel. And so I think educating physicians is something that's really important. And finally, I think this one is the most interesting, is patient care guidelines. And actually the most recent evidence-based patient care guidelines in 2014 actually have different levels of evidence. And so level B evidence is via a cohort of different physicians say that it's a good idea. Level C evidence is that it's an expert opinion. So basically my idea that it should be done. And the importance about this is that level C evidence suggests that exome sequence or even panel-based sequencing be done. And so we really need to change those guidelines because that's not expert opinion anymore. It's really should be standard of care that if you don't know the genetic testing of a patient, that you should do either exome sequencing or panel-based sequencing to identify the genetic abnormality. So in thinking about breaking down test barriers, I really give credit to the Jain Foundation as well as the Muscular Dystrophy Association, where they started offering free genetic testing programs in MDA clinics. And I think this is a great figure where they're able to show the breakdown of the different types of muscular dystrophies that occurred in patients seen in these clinics. And we're able to show the percentage of patients that have this. But this figure here, which is the one that many of us see, is only part of the story. The actual story is that only about 23% of patients fell into that category, and that 77% of patients actually are undiagnosed, and half of them have what we call variants of unknown significance. And so how I think about it is that we broke down some barriers, but we revealed some new barriers and some new challenges to genetic test results. And so what are the challenges of variants of unknown significance? And those are the variants or the changes in the recipe that lead us to not be able to predict whether that cake or that protein is functional or non-functional. And the challenge is that it leaves patients with diagnostic uncertainty. So patients don't know, do I have this disease or not? It allows for the inability to really start to plan for your family and say, I think that this could be inherited. I want to have kids. I don't know. So that diagnostic uncertainty is really important to resolve. It leaves physicians with diagnostic uncertainty. And I put that up, not that we're the most important person, but we really do care to make a diagnosis. And it's frustrating for physicians not to be able to make a diagnosis on the patient's behalf. And most importantly, it may lead to inappropriate treatment and other testing. If I can't tell you that you have a certain disease, you may undergo multiple tests, biopsies. God forbid there is a therapy that you get and it's an inappropriate therapy. Even thinking of a gene therapy approach that could be, even if you have the disease and you don't get the gene therapy approach, but let's say that I tell you you do have the variant and it's causing the disease and you get the gene therapy inappropriately. So that's equally important. So making sure patients do and don't have the different limb-girdle muscular dystrophy disease genes is important. And ultimately, it also leads to increasing healthcare costs and patient frustration. I think this is important too, because we haven't resolved many of these variants. We start to not understand what the true prevalence is of many of these limb-girdle phenotype subtypes are. And I think that's really important because the way that we start to understand resources and engaging drug companies is by making sure they understand how common or uncommon certain subtypes are. And I think I already touched on this. You might ultimately miss out on a certain therapy if we can't make your variant known as being pathogenic. Again, the same thing being said again in a different way. We want to variant resolve because we want to resolve pathogenic variants to determine if people are amenable to therapies, but we want to resolve ones that aren't pathogenic, benign variants, because we want to make sure that a future therapy is potentially contraindicated. And one thing I can also say is by resolving a variant in you as a patient is actually improving patient care for other patients that may have variants or similar variants. And so as we start to do this, it's really something where you as engaged patients can help make a difference for patients in the future or patients that might be less engaged in their care for a variety of different reasons. So it wasn't really until 2012 that we really appreciated the amount of human genetic variation. And it was this this paper here, which was from the 1,000 Genomes Project. And I always think it's funny because it's 1,000 Genomes Project, but they did 1,092 human genomes, but it was called the 1,000 Genomes Project. It was really the first description of what we call rare variants. We already knew that there were variations in families. We'd already been doing that type of studies, in crime labs to understand, even paternity testing. We knew what we call kind of common variation, and if we looked at multiple common variants, but what we didn't know was the amount of rare variants. And what became interesting is that for a single individual, there are many common variants, but there's a small fraction of rare variants, and there's even new mutations or new variants in everybody. And so the way I think about this is that everyone has 3 to 4 million variations. These may be common, but 30 to 40,000 are rare, and some of them are unique to you. And that's not just patients with limb-girdle muscular dystrophy. That's everybody. That's me. That's you. That's everybody in the room has 30 to 40,000 rare variants. Of these rare variants, 250 to 300 are loss of function variants, meaning that I can predict that they're going to cause a non-functional protein. That's amazing to me that within our human variation that we have mutations in genes that cause them to be detrimental. 50 to 100 of those variants are in genes implicated in inherited disorders, and 0 to 7 of them in everybody. So some people have known pathogenic variants in genes, but don't manifest with disease. And so I think it's really, really interesting. And so to me, it really should be no surprise that whenever we do a genetic test, that many of us have variants of unknown significance, because as I said, we have 30 to 40,000 rare variants. Some of those are going to be in limb-girdle muscular dystrophy genes. The other way to think about it is whenever we do genetic sequencing and of patients, and actually put here, actually of any individual, as we go and we take those variants. So we do a patient or even myself, I have 2 million variants, because I just told you that everyone has 3 to 4 million variants. So let's say I have 2 million variants. We then do a filtering process where we look for the ones that are really rare. And so now we end up with that 30,000 to 40,000. We look for ones that actually might be deleterious. We look for ones that we think could functionally predict. And now we're up to 3,000 genes. That's still a lot of variation in genes that could be causing disease. And then we try to enrich for variants that might track with disease. And the point is that everyone has variants of unknown significance, up to 3,000. And so whenever we look at genetic test results like this, and we see, and they've got variants, I very well could have this exact same pattern on my test results. And so the question is, does the patient have progressive weakness? And are these pathogenic or are these incidental? And as we look through these different variants, here's another patient's genetic test results where they have variants within genes and multiple genes. And are these pathogenic or are these incidental? And so this leads to kind of a detour in how we could do genetic testing. And I think of two different approaches. There's a gene first approach, and where we do the genetic testing first, and then there's clinical pathologic assessment. So see the patient, do a history, do laboratory studies, and do that first. And we're going to give both examples, but there's advantages to both. The ease and the low cost of genetic testing has led many patients to receive genetic testing first without clinical assessment by a neuromuscular physician. And I think we need to acknowledge that that's a reality that's happening, that in many cases, gene testing is being done first. And we could imagine a day where gene testing will be done before anyone even sees a physician. And so that's one approach. The lack of experience with genetic testing leads many neuromuscular physicians unable to appropriately order and interpret genetic testing. And so that's the example of going and seeing a clinician, and the clinician sees you, does a clinical pathologic examination, but doesn't really know what tests to order and how to order them. And so I think my point is that we need to be working together. The geneticists who want to order the genetic test first need to be working with the clinicians who want to examine the patient first. And so that's the goal in my mind. The vision is that we not put it on an individual caregiver or physician or geneticist, but that we work together. And so I want to give an example. This is a patient of mine who came into clinic. And he'd already had genetic testing done. And in the case of his genetic testing that was done, he was identified as having a variant in a limb-girdle muscular dystrophy gene that was identified. It was pathogenic. It was in other family members. He had a disease that made sense. He also had a variant in another gene, but that gene was a variant of unknown significance, and he had already a slam dunk made. So he already had a genetic diagnosis whenever he showed up to me, and that was done. This is another patient that showed up to me. This patient showed up to me and said, hey doc, I have limb-girdle muscular dystrophy because I have a mutation in this gene. He has a variant of unknown significance. As you can see, he was a male with developmental delay. He didn't even have muscle weakness. And so my point is that just because you have a genetic test that shows a mutation in a gene, it doesn't mean that you actually have the gene. The geneticist, and so this patient was never seen by a physician, he was seen only by a geneticist. The geneticist can tell you if that gene is rare and unique. They can offer some possibilities about whether it's deleterious. But unless that variant has been identified as pathogenic and is put into a clinical database, it will be a variant of unknown significance. And so this patient needs to be seen in clinic to try to understand if he has a variant, if this variant is pathogenic. Again, I want to give more examples where it just becomes really diagnostically challenging. I don't want to harbor on all of these, but you can see that everybody has variants of unknown significance in multiple genes. This is not just a patient with limb-girdle muscular dystrophy. I want to focus on this example here, where this patient is a 34-year-old man with limb-girdle muscular dystrophy of unknown genetic cause. He had a panel performed, and he was identified as having a pathogenic variant in one gene. And so for some diseases, you need to have two mutations or two variants. For some other diseases, You just need one variant. And we'll talk about that a little bit in a minute. But in this case, for calpain-3, limb-girdle muscular dystrophy 2A or R1, you need to have a variant in 2 different genes. And so he had a pathogenic variant in 1 gene, and he had a variant in another gene. And this one was an interesting variant because this variant didn't change the protein. This was one where I couldn't look at the amino acid change and tell you if it was going to be a good cake or a bad cake. In fact, if I were to look at this, I'd say this isn't going to change the cake at all, is how I would look at this without doing some more work. And so this patient had a variant of unknown significance in this gene. So how do we decide? How do we resolve the variants? What are the steps toward a diagnosis? Well, we try to do what I call an integration of the genetic data and the clinical data. And so we take a patient history. We find out when the age of onset was. We ask what the first symptom was. We see if they have other symptoms, maybe skin changes, heart problems that might make me think of other diseases. Do they have a history of needing a cane or walker in the family? I ask every patient, some of these diseases are late onset. And I find out that the father died at the age of 28 because of a car accident. Well, that's helpful to know. He may not have had muscular dystrophy, but he may not have been old enough to manifest with weakness. Cousins with muscle weakness, that can be helpful. So thinking about distant family members. Physical exam, does he have weakness, contractures, other features? In the case of this patient, his weakness began at age of 25 and his first symptoms were difficulty climbing steps. It's really helpful. That tells me that his weakness is proximal, what I would expect for someone with limb-girdle muscular dystrophy. He didn't have any family history of weakness, not uncommon. In genetic diseases. In the United States, we have small family structures. We don't always know our extended family very well. And so no family history of weakness does not mean that it's not a genetic disease. Proximal weakness was scapular winging. So he had a pattern of weakness and he had this kind of interesting aspect where he had weakness in his shoulder girdle that caused scapular winging. That can be helpful as a clinician. Other aspects we look at, interpretation of laboratory data. So muscle enzymes, electrodiagnostic studies. I would say every patient who could have a muscular dystrophy, we want to make sure they've had an EMG done. Electrocardiogram, do they have heart issues? Sometimes an MRI is done, and often a muscle biopsy is done. In the case of this patient, his muscle enzyme was elevated. It was 2200. He had electrodiagnostic studies that showed that he had a myopathy. He didn't have something like a neuropathy. You can see how this myopathy, this EMG is not telling me anything other than this patient has a muscle disease. It's not telling me what the genetic cause is. We did not do an MRI on this patient. He did have a muscle biopsy. His muscle biopsy showed chronic myopathy with muscular dystrophy. Again, helpful for me to know that he had a muscular dystrophy, but nothing specific telling me that he had a certain subtype. We then go and we become geneticists in some ways. We do database searches. We do functional predictions on those mutations. And remember, he had a mutation in calpain-3. He had 2 of them, and he had one in dysferlin. The calpain-3 is unreported in population databases. With a little bit more legwork, you can actually see that it's very close to what we call a splice site, leading to what's called a donor loss. So it could lead to a truncated protein. That dysferlin variant, although he only has one dysferlin variant, is rare, and it has a minor allele frequency of 0.03. This is one of those 3,000 rare variants that this patient probably has that may or may not be pathogenic in this patient. The other things we do is we say, well, let's expand the patient's pedigree. Are these mutations on different chromosomes? I don't know that from just doing a single patient. And so we expanded the pedigree and we found out his father was actually had already passed away, but his mom had one of the mutations in the calpain-3 gene. His sister was a carrier, and the other sister that was unaffected. So these were unaffected patients, and it would make sense they were unaffected because they didn't hold both those variants and both those mutations. We were able to do immunohistochemistry on his muscle biopsy. The immunohistochemistry, in some cases, this showed abnormal dystrophy-related protein stains with reduced dysferlin. Well, that makes me start to think that maybe this patient doesn't have mutations in calpain, but actually has a mutations in dysferlin. We need to go the step further because he only has one mutation in dysferlin. And what we do is in our lab, we do an extra step where we actually do a Western blot to actually look at how much calpain-3 protein is there. And you can see this patient has absent calpain-3 protein, but normal levels of dysferlin. And so in this case, we solved this variant is resolved. This patient has Limb-Girdle 2A, R1 calpain-3 associated. And as I hope you can see, this was time consuming. This took a specialist. This took me thinking about this patient. This required acquiring patient family structures and DNA. This required specialty testing. And that's what it takes sometimes to resolve these types of variants. And I can't expect non-specialists, and I can't even expect some neuromuscular physicians to be able to do this process. And so I think it's really important to appreciate that. I bring this up because Brad Williams asked me to make a comment on this, and he said, you know, some limb-girdle genes are even more complicated. And so I put here the complicated becomes even more complicated. And so some limb-girdle muscular dystrophy genes can cause a disease in a recessive manner, meaning that you need two disease mutations, one from mom and one from dad. Some limb-girdle muscular dystrophy genes cause a disease in a dominant fashion, meaning that you just need one disease mutation and you can get that variant from mom or dad. But the complicated gets more complicated because we're learning now that some limb-girdle genes can be inherited in both manners, in a recessive or a dominant fashion. And it makes the challenge of variant resolution very complicated. And I don't have a clear answer for how to solve that problem. And likely it's going to require just more sequencing of patients and families, and more looking into variant and mutation specific variation in patients. And so hopefully the process I'm going to describe of how we'd like to look at more variation will help. We've become to be part of a group called ClinGen. This is an NIH supported infrastructure. It does exactly what we said. It's systematic variant resolution. And so it integrates patients, clinicians, genetic testing labs, and researchers to really ask three fundamental questions, which is a gene associated with the disease? Is that variant causative for that disease? And really the one that I think will become the most important is that information clinically actionable. And we think of this in cancer genetics, where if someone has a BRCA1 variant that's known to be pathogenic, that they need to be actionable. And hopefully someday, if you find that you have a variant in a certain limb-girdle muscular dystrophy gene, this will be a clinically actionable, meaning that you will need to know that there's a therapy out there for that disease. And the way that we do this is by a very complicated approach where we look for levels of evidence and try to integrate in population data, functional data, segregation data, all the stuff I just described to you, and try to do that in a systematic way and kind of build levels of evidence and strength. And I like this slide because they're Lego blocks, and what we're trying to do is build them and stack them and actually have variant classification and ultimately lead to what we call expert panel reviewed. And so I finally just want to summarize here that at present, genetic test interpretation, or what I call armchair variant resolution, really requires knowledge by both clinicians and geneticists in muscle disease. Systematic variant resolution, which is what we'd like to do, is really, I think, the way to go, so that basically we can move into a gene-first approach where anybody can get genetic test results and they'll know if it's a pathogenic variant. And finally, what I call enhanced clinical phenotyping, it needs to be done by a limb-girdle muscular dystrophy expert to really understand a genetic diagnosis, and that integrates all of these aspects into kind of this stacked level of care. And I want to thank members of my research group. I want to thank a large LGMD research consortium, which is helping us understand these questions as well. So thank you.
Hello, my name is Kyle Harrington, and I'm from Illinois in the United States. And I have limb-girdle muscular dystrophy 2B, also known as R2, also known as dysferlinopathy. My diagnostic journey began in 2001. after giving blood at a school blood drive. I had finally turned 16 and I was so excited to give blood. It was something that I always wanted to do, to give back to my community and people who may need it. So I was so excited, gave blood, and a couple months later got a letter from the Red Cross which said I had an elevated liver enzyme and I probably should go get it checked out. So I started my first of many, many, many doctor's appointments. They couldn't really tell what was going on. I had one doctor who, again, I was 16 years old, and one doctor who accused me of being a secret drinker and kind of all of these crazy things that might justify this elevated liver enzyme. So 2 years later, after several tests, including things like a mammogram, colonoscopy, endoscopy, gallons of blood given, I finally discovered that I had an elevated CK level, which was highly elevated, not just, a little elevated. We're talking like 15,000 when it should have been about 100. So after that, with the new information of this elevated CK level, I began to see a rheumatologist. They did a few tests that really kind of, you could tell that something was going on, but it wasn't really, really clear. I was still, in really good shape, was an active athlete. I did musicals, I did all sorts of stuff, active stuff in high school. And, but they noticed I couldn't stand on my toes and I couldn't get up from the floor quite as quickly as, your average 16-year-old probably should be able to do. So they did more tests and couldn't really figure out what was going on. We had no family history, no real clue. So I went down to Barnes at WashU in St. Louis, and surprise, surprise, more tests. They did an EKG, they did all sorts of things that, I cannot even remember anymore. But one of those things was a muscle biopsy. And when the results came back, it kind of looked like there was something, going on. I could see some scar tissue, and they thought, well, this doesn't really look normal. So we're going to go with what's called polymyositis. And that was my first diagnosis. Polymyositis is an inflammatory muscle condition, and it's treated with prednisone. So from there, I was put on prednisone, which the doses got increasingly higher as I didn't really show any improvement. And it turns out that I would soon have a very, very steep decline. Any side effect that you could have with prednisone, I got. So I had steroid-induced myopathy. So my legs would just give out from under me when I was walking. I couldn't walk upstairs anymore. I certainly couldn't run, couldn't jump. I got acne. Like early signs of depression, a host of really horrible symptoms, which, ended in me having to leave school because of these symptoms. I was a sophomore in college at this time. So it was a pretty scary time when you just want to be with your friends, hanging out, you're going to school and being like any normal 19-year-old. I was going through this, pretty trying time, and had gotten so bad that my parents thought, we're going to have to figure out how to make our home wheelchair accessible within just, now 2.5 years post the Red Cross letter that I had an elevated liver enzyme. So it was a really, really roller coaster of a couple years. Once the prednisone showed that there was no improvement at all, and again, a very, very steep and pronounced decline, my muscle biopsy was tested again using what's called a Western blot stain, and they discovered the absence of the gene dysferlin, or the protein dysferlin, in my muscle biopsy, which was the first indication that something bigger was going on. So because of that Western blot stain, they unofficially diagnosed me with dysferlinopathy, or Limb-Girdle, now R2, also known as 2B. And that was that. I was able to go off the prednisone. Although I never got back to kind of where I was before being on prednisone, I lived a very, very, very normal life. In 2011, I finally got my first genetic confirmation of one abnormal gene. And then it wasn't until 2019 that I finally got my second confirmed mutation. And, now I am official LGMD girl. So that's my story. It's been a long road. I mean, now 20 years post-diagnosis. And it was long and it was arduous. But, here I am. So thank you very much.
Hi, my name is Jenna Keindel. I'm from the small town of Shawville, Quebec. I'm excited to share my story and the ups and downs of my diagnosis journey. My LGMD story started to take shape when I was 16 years old. That's when people around me began to notice I had some muscle weakness. I was having trouble getting my legs into the car, and I couldn't jump for the net in gym class or throw a baseball. I was having a hard time with stairs and couldn't reach over my head to put my hair in a ponytail. But even before all of that, I can recall not having the ability to do sit-ups and push-ups with my classmates, even as a child. After several assessments by PTs and OTs and being referred to the Children's Hospital of Eastern Ontario to do an EMG to isolate the muscles that were affected, the specialist concluded that I had a form of muscular dystrophy. More specifically, I had limb-girdle muscular dystrophy. The next test would be more invasive, and they would need to take a muscle biopsy of my bicep to confirm the diagnosis. So in 1997, I had my first of 4 muscle biopsies over the next 23 years. Doctors and researchers ran my DNA and then reran the DNA tests several times over the next couple of years. I looked at my parents' DNA to see if any indicators of what subtype of LGMD could have been passed on genetically. I had my genome sequence done in 2014 and still no significant signs to point doctors to what subtype of limb-girdle muscular dystrophy I had. I had a massive family, at 15 aunts and uncles and over 50 cousins, but not a single relative with a muscle condition that we knew of. One of the things that I knew for sure was I was getting weaker, and we still didn't have the complete diagnosis. Shortly after turning 32, I gave up my driver's license because I didn't feel safe controlling and steering a vehicle anymore. My legs wouldn't cooperate the way I needed them to be able to brake and accelerate safely. I wanted answers, and I wanted to fit into a group. I wanted to sign up for clinical trials and research groups. And more importantly, I wanted to qualify for treatments if and when something became available. That was the most important reason that I wanted to keep going and looking for my genetic diagnosis. So part of having a rare disease, we learn how to cope and adapt all the changes. I like to turn to social media because Facebook has several amazing closed groups for limb-girdle muscular dystrophy. Having friends online and peer support groups that I can turn to 24/7 is a great outlet for conversations, and there's always people online to bounce questions and ideas off of. These groups are full of people who just get me. They connect thousands of people from around the world. And another great thing about these Facebook groups is you don't even have to post and comment. You can just be a fly on the wall and just read and relate. Or you can vent and post something that's been bothering you, and the rest of the world sometimes just doesn't understand. But the members of the group are there, ready to shower you in support and provide words of encouragement. Another great thing about these groups is the ease of sharing information. There's a lot of links and documents that are getting posted, and I try to stick and only read the articles from reputable sources that have all been proven to have, the right research done and the data to back them up. In January of 2019, Brad Williams shared a scientific article from the NIH titled Anti-HMG-CR Myopathy May Resemble Limb-Girdle Muscular Dystrophy. In Brad's post, he added that it was something to consider if a person had symptoms consistent with LGMD, but genetic tests hadn't found any leads for yet. I was intrigued, so I printed off the article and sent it to my doctor and asked her to review it for our upcoming appointment scheduled. When I went to see her a few weeks later, we discussed the possibility of an autoimmune disorder, but I was also telling myself to be conservative with my hopes and my expectations because, after all, I was a poster child for having all of the characteristics of LGMD. My doctor sent me for blood work. It was 3 weeks. It was going to take 3 weeks to get the results back. They ran a panel of tests looking at several antibodies. 3 weeks later, I received a call from my doctor's office, and my specialist wanted to see me in her office the next day. So I went in. My blood work came back highly positive with levels of anti-HMG-CR myopathy antibodies. We were both shocked and surprised by the findings and decided to run the test again to be sure that it wasn't just a lab error or a false positive. So I rolled up my sleeve and I gave another vial of blood for another test. 3 weeks later, my specialist called to tell me that she had shocking news again and was confident that I didn't have limb-girdle muscular dystrophy and that my diagnosis was confirmed to be anti-HMG-CR myopathy, an autoimmune condition, a disease that would have never shown up on any DNA test. In closing, I'd just like to stress and remind you, encourage you never to give up hope in finding your diagnosis. Keep pushing for that label. It took us 23 years to get to the bottom of it for my diagnosis, and research is constantly evolving. I'm sure doctors have a hard time keeping up with the data and the articles that are being published. So if you're still on a journey and your doctors haven't been able to pinpoint your subtype just yet, and you've had your biopsy done, go to your doctor and ask if they'd consider other conditions that present the same way as LGMD. It's a single blood test, and the test will come back one of two ways. It'll either be negative, and that's also part of the puzzle piece, or it'll be positive. And if it's positive, then you'll have to look up my new Facebook group and join our peer support group because we have a lot to talk about in and about HMG-CR. In June of 2019, I started my treatment process, and it involves intravenous immunoglobulins, which is IVIG, and these involve connecting to an IV port and sitting at the hospital for hours every 2 weeks. I'm also on a cocktail of immunosuppressants, chemotherapy, and steroids to help slow down and stop my immune system from doing more damage. My hope is that I'll gradually regain some of my strength. I've set some small goals for myself that I hope are realistic, and little by little, I hope to accomplish each one. I've already noticed a few improvements, and I know that I can flick the light switches on a little bit easier, and I can turn in my bed. I can move my legs when I'm laying down. I even bought a brand new van in 2019 and got my driver's license reinstated. I have adopted equipment that helps me drive with confidence. I can control the vehicle, so it's nice to have that independence back. I've heard of other people who have the same autoimmune disorder as I do, who were once using wheelchairs, who now have the ability to get up and walk again. I'm not quite there yet, but I'm working towards that goal. Right now, I'm enjoying my aqua physio and doing lengths and lunges in the pool. And maybe one day I'll have the strength to do my own transfers independently. Perhaps I'll be able to stand up and reach for something on the top shelf of the bookcase one day, who knows? I'll count my blessings and thank the researchers for all of their hard work and their dedication. Again, I just want to encourage you to talk to your doctors if they're still searching for a subtype. Ask them to consider running a panel for antibodies to see if there's any red flags. You might just be as surprised as we were.
Our last speaker is Dr. Monkol Lek, a PhD researcher who is an expert in genomics and genetics from Yale School of Medicine in New Haven, Connecticut. Over to you, Dr. Lek.
Thank you, Laura, for the kind introduction. And I'd like to thank the organizers for giving me this wonderful opportunity to talk about something that I feel very passionate about, and that's genetic reports and what patients should do if they have a non-definitive genetic diagnosis. And it's something for patients to understand that just because you got a genetic testing, that not all results are actually definitive. So I'm going to go over, as an overview of this presentation, different types of genetic testing you can get, and how you actually go on interpreting genetic reports, particularly in the case where there is a non-definitive genetic diagnosis. Now, understand that I first talk about inheritance patterns and then what is and isn't a definitive genetic diagnosis. How do you interpret that report without a definitive genetic diagnosis, and what to do afterwards, which is also very important and should sometimes be driven by the patient and what they want to actually do afterwards. And I'm going to briefly touch upon one of my research focus, and that's the process of discovering new disease genes. And lastly, end off with 3 genetic testing programs. I'll talk about 2 examples, but it's not an exhaustive list, but just 2 examples of some of the goals of these genetic testing programs. So first of all, to talk about genetic testing itself. So there are 4 main types of genetic testing, and they and the difference is how much they actually cover. So the first is single gene test. So this is just sequencing one gene of interest. And this is typically done when discoveries are made in a research setting and they need to be validated in a clinical setting. So we're either sequencing just one variant in the gene or the whole gene itself. And this is typically more sensitive because these are guaranteed to have more coverage. And a single gene test can also cover things that are not genetic. So we can look at protein levels, we can look at protein localization in a muscle biopsy, and we can also look at enzymatic activities as a way of doing a single gene test. Another and a very popular way of actually doing genetic testing, a gene panel. So this is a set of genes, typically 100 or 200 genes known to be associated with neuromuscular diseases. And for the focus of this talk, limb-girdle muscular dystrophy. So the idea behind this is that we know that most of the limb-girdle muscular dystrophies are explained away by just a handful of genes. So in terms of the recessive ones, these are your calpain 3, dysferlin, and also FKRP. So the idea is, let's go for the most obvious genes first. And this is a very cost-effective way of actually doing it, and it reduces the burden on the analyst that is actually going through the results. The next is exome sequencing. So exome sequencing is looking at all the genes, all 20,000 genes, and not just a subset. So we're not sort of like, we're not limited to a particular hypothesis that a known gene is actually causing your disease, or that the symptoms you're presenting are the only symptoms, and also the biases of picking that. So there are symptoms that are overlapping with limb-girdle muscular dystrophy that may not be in that gene panel yet, and also novel disease genes. And last is whole genome sequencing, which is doing the whole watermelon. Exome sequencing only covers about 2% of the genome. This is the region of the genome we best know how to interpret. The other 98% and the variants we see from it, we have a much difficulty in actually interpreting, and this is something that is atypical that we would get for a patient, and it's more still in the domain of research. So there are the types of different genetic testing, and a lot of that is empowered by this technology called next generation sequencing, allowing us to sequence lots and lots of DNA fragments simultaneously, and therefore the power of actually being able to look at, say, 20,000 genes at once. However, with next generation sequencing technologies, this uses a short read sequencing, and so there are classes of variants that we can pick up easily and some that we have great difficulties. So the things that most of these genetic tests can pick up are single nucleotide variants. So these are just changes in the DNA letter, so from, say, an A to a T. So it's just a single change. A deletion, this is deleting just small bits, so like a few letters, or insertion of a few letters. So next generation sequencing does a fantastic job of actually detecting these variants. And the great thing about these variants is that they account for most of the mutations that we actually see in limb-girdle muscular dystrophy. But however, it doesn't cover all the mutations we see. There are also tandem duplications, interspersed duplications, inversions and translocations, and also copy number variants. And we consider these more complex types of structural variants. So some of these we can kind of pick up, but we're not very powered to detect it. So there's a high false positive and negative rate, especially with copy number variants also. So this is the pros and cons of each genetic test and what they actually cover. So more specifically, the diseases that they struggle to detect, a lot of these genetic tests, are FSHD, which is actually caused by a repeat contraction of the D4Z4 repeats, myotonic dystrophy caused by repeat expansions in genes, in two known genes in the intronic region. So introns are typically not covered in a lot of these genetic tests, only in genomes, and even with short read sequencing technologies, we have a hard time detecting repeat expansions. And the last is spinal muscular atrophy. So the genes underlying the disease mechanisms are highly identical. So short read sequencing technologies have great difficulties in telling the difference between these. So typically with these three tests, people actually order tests that are more sensitive to actually detect these three diseases along with a gene panel and things like that. So sometimes they come with the gene panel and sometimes they come additional that your neurologist will actually ask for. But it's just to say what the limitations of these technologies are and approaches. So next I'm going to go into interpreting the genetic test itself. So to understand the interpretation of genetic tests, we need to first understand the typical inheritance patterns that we see in limb-girdle muscular dystrophy. So one inheritance pattern we see is autosomal dominant. That is where one of your parents is a carrier and is affected by limb-girdle muscular dystrophy. And in dominant cases, you only need one mutation in a diseased gene that causes limb-girdle muscular dystrophy, and there's a 50% chance that this will actually be carried by the offspring or the children of this parent. And so these are normally labeled with the new nomenclature LGMD, D for dominant. A subset of these, but something that happens very rarely, is what we call a de novo dominant mutation, and this is when the parents are not carriers and the mutation actually occurs in the egg or the sperm, and therefore the next generation can carry this mutation. And so the mutation again is a single mutation in a gene known to cause dominant limb-girdle muscular dystrophy. Another common, so this is less common, another common form is an autosomal recessive, and that's where your parents are carriers, so they have one defective copy of a gene, but they don't actually have the disease, unlike autosomal dominant. And there is a 25% chance, so 1 in 4 chance, that the children will carry 2 defective copies and then be affected by a recessive form of limb-girdle muscular dystrophy. And these are typically called LGMD, so R1, R2, etc., etc. So knowing the inheritance patterns is important by going when looking at the possible outcomes in a genetic report. So one possible outcome, and this is where patients are happy they've actually gotten the answers, is definitive genetic diagnosis. So in the case where a suspected recessive pattern is assumed or thought to be the case in a recessive disease gene, and I've bolded and underlined this for a reason, because it's important that these things actually happen. And in the genetic report, that there are 2 variants. We need 2 variants, so both copies need to be defective in a recessive case, and they're also reported as pathogenic and likely pathogenic. So this has to occur to be a definitive genetic diagnosis for limb-girdle muscular dystrophy. And typically what we also look at is we actually confirm that the parents are carriers of the pathogenic variant also. So going back, we want to see that one parent is a carrier of, say, mutation 1, while the other parent is the carrier of mutation 2, and therefore the child receiving 2 defective copies. So in the other scenario of a dominant disease gene in limb-girdle muscular dystrophy, we're looking for 1 variant, but however, it still needs to be reported as pathogenic or likely pathogenic in a gene known to cause a dominant form of limb-girdle muscular dystrophy. And typically 1 parent is a carrier and is also typically affected. So what will typically happen is, if it's not known, that the parent will be re-examined, because sometimes what we see in dominant diseases is 2 concepts. Incomplete penetrance, so sometimes people don't get the disease or get the disease later on in life, or variable severity. So some people are so mild they don't even realize they have the disease. So, and that's why it's important also to actually re-examine. So now that's a case of a definitive genetic diagnosis. So what does a non-definitive genetic diagnosis look like? And it's very important to look at your genetic report and revisit it if you have it. So one is in the case of a recessive disease gene, this is typically when only 1 variant is reported as pathogenic or likely pathogenic, while the second variant could be either reported as variant of uncertain significance, and this is covered by another talk, so I won't go into the details, but some examples of ways you can actually resolve and actually provide more evidence is, say, ordering clinical tests such as the MRI and looking at the muscle wasting pattern and whether it's actually typical for your limb-girdle subtype and also gene associated with your limb-girdle muscular dystrophy. The other possibility is the second variant is not reported or detected. And this is what I'm going to call scenario 1. And later in the next slide, I'll go into the details of what that actually means. The other possibility, if you have a dominant disease gene, is that the variant is actually reported as a variant of uncertain significance. Again, this will be covered by another talk. The possible ways that clinicians and researchers actually go and resolve and a variant of uncertain significance, hopefully into either a pathogenic or a benign variant. And the last possibility is all variants are reported as variants of uncertain significance, or VUS, or no variants are reported or detected. So this is scenario 2, and this happens more common than you think. Just a reminder that only 50% of patients actually get a definitive genetic diagnosis. One thing I'm going to have to stress, because I've talked to a lot of patients showing their genetic report and coming to this conclusion, and I just want to elaborate on this. Multiple variants of uncertain significance reported across different genes does not mean you have multiple muscle diseases. So I'll put this in context. A variant of uncertain significance is a rare variant in the population. And what I mean by that is that not many people in the population has that exact DNA change. But to put that in context, when you screen a lot of genes, and also particularly large genes, which muscle disease genes are, by chance, you're going to start seeing some of these rare variants that are difficult to interpret as pathogenic or benign. And also, just to let people know that even healthy people have rare variants also. It just means you have a DNA change that's not common in the population. And these changes for even healthy people can happen in any gene, and that includes disease-causing genes too. They're called variants of uncertain significance because we're not quite sure if it causes disease or if it's benign. So just to put that in context, that even healthy people, if we did screen them, we would see something like this too. So I'm going to go over scenario 1. So scenario 1 is a recessive disease gene where the second variant is not reported either. It's not reported as pathogenic or it wasn't detected at all. So I'm going to go over possible next steps or alternate conclusions. One is we could do a gene-specific test. So we could do sequencing specifically for that gene, getting higher coverage with the ability to detect different types of variation. Or we can look at supporting gene-specific tests that aren't genetic, looking at protein expression, the localization of the protein, for example. An alternate conclusion could be that you're a carrier of a common disease mutation. So the FKRP founder mutation that I've listed here could be as high as 1% in the Northern European population. So you could just be a carrier of this mutation, and there is no second mutation because there is another gene that's actually causing your disease. Another possibility, next step we could do is analyze the data for copy number variants. So typically the data generated by this is not typically analyzed by clinical genetics labs because of the high false positive and false negative rates when looking at copy number variants. So we could either use the gene-specific test or use research methods to actually look at that. We could also reanalyze the data for variants that are typically not interpreted. One such variant is a splice donor position -1 variant. This is not going to mean much to the patients in the audience. This is more for researchers and clinicians to know that sometimes reanalysis can pick up extra variants, and this is what we typically find in our research. You could also request exome genome sequencing. Typically, you would request exome sequencing for cases where you've only done a gene panel to look at the remaining genes also. And so not necessarily look for that missing variant. And the last is, but genome sequencing is typically not ordered. And the last is enroll in research study where we actually apply the cutting edge genomic technologies to actually look for either the second variant or another possible gene. So we perform either genome sequencing, RNA sequencing, and the last is long read sequencing to detect difficult to detect variants. And that would encompass some of the variants that I actually mentioned in FSHD, myotonic dystrophy, and also SMA. So long read sequencing can detect that. The only problem with long read sequencing is it's high cost and also still high false positive rates due to low coverage. So it's still in the research realm trying to make that technology better, but it holds that promise. While in scenario 2, where you have no variants reported as pathogenic, one such case that patients and their physician need to revisit is that, is the disease actually even genetic? So we have a patient in this session that will talk about their story in terms of this. Next is, if you only had gene or gene panel sequencing performed, then you can request exome sequencing. So this will remove us away from the bias of, your symptoms and what they could be to just being hypothesis free and searching for all possible genes, because we know that a lot of diseases have overlapping symptoms. So it allows us to detect either novel disease genes or genes that we would not initially think of that wasn't present in the panel. The last is we can actually enroll patients in research studies. So this is what I'm highly involved in my day-to-day research, to find possible novel disease genes, but I haven't listed here, and also novel mutations and disease mechanisms in known disease genes. And it's just a reminder, this is an active area of research because we only have a 50% diagnosis rate in rare disease patients, sometimes a little bit higher, sometimes a little bit lower, depending on the disease and the mutations we typically find. So in these research studies, we use new genomic technologies and also analytical methods to help solve the remaining. We can also apply novel analytical methods on new or existing data. So hopefully that sheds light. And the reason why we're able to, achieve success in discovering novel disease genes is we recruit a lot of patients with the same disease symptoms. And so because to associate evidence, we require multiple patients to be sure that the candidate pathogenic variant and novel disease gene is the right one, instead of only having an N of 1 or one example. And what we do to strengthen that evidence of a discovery of novel disease gene is we normally create a mouse model or we create another animal model. So for neuromuscular diseases like limb-girdle muscular dystrophy, zebrafish models are also popular given their reduced cost and speed in actually generating them, and their similarities in terms of muscle structure. And so having all that, we could conclude a novel disease gene, which eventually will be added to the gene panel. So that's how research goes about in addressing scenario 2. So just to touch upon people that fit in scenario 2, the best way of actually getting in contact and being part of these research studies would probably be to talk to, first of all, your physician, and then also reach out to researchers that you know are in this field, and also your associated disease foundations. So I'm going to touch upon, in the remaining minute, 3 genetic programs and 2 examples. The first is the Lantern project, where they perform gene panel sequencing, and this actually replaces the Emory Genetics Lab gene panel, and this is performed by PerkinElmer, where they perform clinical gene panel sequencing of all the genes known to be associated with limb-girdle muscular dystrophy and the ones that are very similar to limb-girdle muscular dystrophy. And this is good for patients that have not performed any genetic testing. And this is a good way of actually picking out people with common causes of limb-girdle muscular dystrophy. However, if you've had some genetic testing, either a panel, gene-based testing, it's unlikely to change the interpretation. Normally, these technologies are already really good. There is a possibility you may detect something new, but normally this technology is really good. And also the interpretation clinical labs perform are very good also. So it's very unlikely to change that. And the last is the Rare Genomes project, where they perform whole genome sequencing. So this is performed by the Broad Institute. It's a research-based genome sequencing. So any findings have to be validated by the single gene testing I mentioned. And so this is good for patients that already had exome sequencing and they haven't had any conclusive diagnosis. And this is unlikely to change the interpretation of variants already reported, like what I mentioned from the Emory genetics. So with that, I'd like to conclude my talk, and and hopefully this is a good refresher of what, say, a genetic counselor went over with you. And it's always good to have a refresher, because when you get that genetic report, it is quite overwhelming. And this is a refresher for you to look back at your genetic report. Thank you.
Hi, my name is Mark Barnett. I live in Michigan in the United States. And I am 60 years old, and my story's a really long one trying to get diagnosed. It all started with my father, actually. When I was a toddler, about 1963, he started having falls and difficulty getting up and climbing stairs. And this was all a mystery to everybody because there had never been any family history. So he was finally diagnosed with LGMD. It was just a very generalized diagnosis based on his clinical presentation. And I was so young, I don't even remember my dad starting to experience issues. He just had it my whole life. So I remember about the time I was 6 or 7 years old, my mom put me in the car one Sunday afternoon, and we went for a ride. She bought ice cream, and we parked at a park, and she explained to me that my dad would be getting very sick, and he probably wouldn't be getting better from this, and doctors weren't sure what caused muscular dystrophy. At the time, they really thought it was some kind of an enzyme that ate the muscle tissue away. That's all they knew in the late 1960s. And that I really wasn't going to have a dad that would play baseball with me or things like that. But he'd probably be along for a long time. They figured he'd be around till I was quite a bit older, so there was no real danger of me losing him within the next few years. So years went by. I was a slow child. I was always the slowest kid out of my friends, slowest kid in gym class. I would play sports, but I was always the last one to come in, last one to get picked. That's often the case with LGMD patients. But pretty much it was normal. I played basketball, I played golf, I played softball in my 20s and my 30s, and things were pretty normal. I just wasn't really super athletic. I was married at the age of 26, and we had 2 little girls, and just enjoying life for a while. And about the time I was 37, 38, the symptoms started with me. I had trouble climbing stairs. It was getting really hard to run. And I knew it wasn't a huge shocker to me because based on the fact that my father had this, I knew I had something. So I made an appointment with a neurologist. I went and saw him. He did a full physical exam. This was 2002, January. And his first impression was, he said, "You certainly have something. You're not able to get out of a chair or climb stairs without your arms. This is something you should certainly be able to do at 38 years old. So there's definitely something wrong. And I'm going to say you have muscular dystrophy based on the fact that your father had it. You've got the same symptoms." So based on that, I told him I would like a second opinion. I'd like to contact the MDA Association and go to their clinic, which I did. And I met a great doctor who would be with me for the next 15 years. When I first went, we did a few genetic tests. There weren't many, only 5 or 6 LGMDs were available for genetic testing at the time. Those all proved negative. So I began a letter writing campaign. I wrote to Ohio State, Iowa, NYU, Duke, several researchers that are actually quite well known in the MD community. And every one of them was great. They wrote back. They offered to look at my blood. They offered to look at my biopsy samples. And we began a lot of testing. I was just mailing letters and tissue and blood all over the country. Everybody had the same conclusion. You definitely have muscular dystrophy. The biopsy shows really severe muscle deterioration, but we just don't know what you have. Everybody else saw the same other mystery, that I had low dystrophin levels, but not an absence of it. So we could pretty much rule out LGMD2B. And this was a mystery to everybody why that was going on. And about 15 years had passed. I did not have a genetic diagnosis yet, and I was in a wheelchair by now. So by 2017, I couldn't walk, and nobody could tell me why. I was determined to figure out why. I started an online group. I called it Muscular Dystrophy, Subtype Unknown. I thought I'd get 20 patients. I had 1,000 the first week, or not the first week, but I had 100 the first week. By the end of about 2 years, I had over 1,000. Everybody had a muscular dystrophy, and they didn't know why. My campaign continued. I actually, through the Jain Foundation, I got my first genetic panel done, 35 genes. That came back negative on everything. I had a variant of unknown significance. It really didn't pan out to be anything. This was kind of heartbreaking and frustrating for me, but I didn't give up. I kept up with my letter writing campaign. Finally, through Brad Williams, I met Monkol, who actually hooked me up with a researcher at Harvard, Dr. Beggs at Beggs Lab. I was able to send blood into him. He did whole exome sequencing, sequenced my whole DNA. He called me 2 long years later, and he said, "Mark, I found your problem. We found a defect in the LDB3 gene, and I am sure this is what you have. It's only a few patients have it, maybe a couple dozen that I've heard of, only a few in the United States. But he says your symptoms are exact, And I am sure this is what you have. So since April of 2019, I finally know why I have muscular dystrophy and why I'm not making muscle. It was a long, long journey, 17 years, but all I can say to people is please never give up, never stop writing letters, don't take no for an answer. My gene is actually on the gene panels now since 2019, and it wasn't. We would have never found it before then. So just never give up, keep trying, don't take no for an answer. You will get an answer eventually.
This brings our third day of the conference to a close. Thank you all for your participation, and thank you to our platinum sponsors, Sarepta Therapeutics, SBio, and GFB Onlus. Join us tomorrow afternoon at 12 p.m. Eastern as GFB Onlus shares about progress on treatments for LGMDs, particularly LGMD2E, their area of focus. We will hear from our Neurologist of the Year, Dr. Yvonne Torrente. As the motto of one of our LGMD advocacy foundations says, together we are stronger. See you all tomorrow.